Hello, welcome to all of our attendees who are joining us this morning. Thank you for being here for this event. Good morning to our West Coast colleagues. Happy lunchtime to those of you on the East Coast, and good evening to those of you joining us from Europe. I have just a few housekeeping notes to cover before we get started today at the top of the hour. So first of all, we want to know who's here and who's joining us. So please take a moment to introduce yourself in the chat. Please share your name, affiliation, and where you're calling in from. And feel free to let us know what you're excited to learn about today or why you registered for the event. Be sure to send your chats to all panelists and attendees as noted on the slide. And to those of you who are just joining us, please introduce yourself in the chat with your name, affiliation, and where you're calling in from. We'd love to get to know our audience a little bit. Hello, Susanna from Nashville. Let's see, Victoria from Jupiter, Florida, welcome. Simona from Seattle, hello neighbor. And yes, as Christina noted in the chat, be sure to send your chat to all panelists and attendees. You can select that from the drop-down menu that's shown on the slide. Again, welcome to all of you who are joining us. We'll get started today. If you open up the chat panel from your Zoom taskbar, send a message to all panelists and attendees with your name and where you're calling in from. Morning, Emil from Minnesota, welcome. Claire from University of Sheffield, hello. Okay, so again, please let us know your name, where you're calling in from, and affiliation if you'd like. Send that in the chat. As we are getting closer to getting started, we are planning to begin right at the top of the hour. I have just a few more housekeeping notes to go through before we get started. So as we get started, please be sure to share any questions that you have for speakers in the Q&A panel. We won't be using the chat for questions. We'll use that Q&A panel. That's also accessible from your Zoom toolbar. Once you open the Q&A panel, you can click to type your question in that window. We ask that you begin the question with the speaker's name so we know who it's being directed to, and be sure to click send. We do have time scheduled for presenters to answer questions from the audience after each presentation. Some will be answered live and some will be typed after the presentations. If we do type an answer to your question, you'll be able to see that by clicking over to the My Questions column of the panel. And answered questions from other attendees will appear in the All Questions column. If you're having any connectivity issues, we have two options for you. The first is you can view the event stream on YouTube by clicking the option at the top of the Zoom window as shown here. You are also welcome to call in from your phone for better audio by clicking Switch to Phone Audio and following the prompts to dial, available in the Audio Settings panel. So here, our next slide, we have a brief overview of our agenda. We will have some brief introductory remarks followed by 4 talks. Then we will have a brief break from 10:30 to 10:45 a.m. Pacific time. Then 2 more talks before our fireside chat. We do hope that you can stay for the entire program, but if you do step away, we will be posting a full recording of the event on the event website and our YouTube channel after the event ends. So now that we are at the top of the hour, I am going to pass it to our moderator, who I'm happy to introduce. Kathy Richmond, Director of the Paul G. Allen Frontiers Group. Please feel free to share any welcomes or hellos for Kathy in the chat, and continue to introduce yourself if you haven't done so. I'm going to welcome Kathy.
Welcome. Thank you, Megan, and welcome everyone to the Exploring Frontiers, the Future of Brain Health Research. My name is Kathy Richmond, and I'm the director of the Paul G. Allen Frontiers Group, a division of the Allen Institute. We are delighted to have you virtually with us for this symposium to envision where the field of brain health research could and should be in the next 5 to 25 years. Before I speak about the event program, though, I'd like to tell you a little bit about the Allen Institute and specifically about the Paul G. Allen Frontiers Group. Now, many of you may know that the Allen Institute was launched in 2003 by our visionary founder, Paul G. Allen. And the Institute was built on the core principles of big science, team science, and open science. And our colleagues take on large-scale foundational questions in bioscience and tackle them with multidisciplinary teams, sharing the data and resources generated openly with the wider community. Now, the Allen Institute for Brain Science is known worldwide for the public resources that they've shared, which include gene expression maps of mouse and human brains. And in 2014, we became the Allen Institute with the launching of the Allen Institute for Cell Science. And in 2018, this followed with the Allen Institute for Immunology also being launched. And these three divisions all operate in Seattle in the South Lake Union region. Now, the Paul G. Allen Frontiers Group, our division, extends our founders' commitment to tackling the toughest questions in bioscience and beyond. And it really stretches beyond the Institute walls by directing funding to investigators and centers across the country and internationally. And at this point, I'd be remiss if I didn't congratulate our Allen Distinguished Investigator, Jennifer Doudna, and her collaborator, Emmanuel Charpentier, on their Nobel Prize Award in Chemistry today. It's an exciting example of frontier science research. Now I'm going to play a brief video to help describe what the Frontiers Group is, what we do, and what our impact aims to be. I hope you enjoy it.
You know, one of the easiest things that people can say to rebut something is, it's never been done, it can't be done, until it's done. So how do you get people to believe in something that, yeah, it's never been done? But imagine we'd never been to the moon, right? Antibiotics. Fleming threw away his Petri dish. Where would we be with disease? So it's really important, I think, to believe, because that's what advances the human condition. It's individuals who've had the courage to say, we need to do this. I don't know what I'll find, but I'm going to do it anyway, because that's how we advance.
The Paul G. Allen Frontiers Group seeks pioneers, seeks explorers, seeks people who are willing to make big advances, take risks, ask questions, be tenacious, and have curiosity to change the face of biology. The Frontiers Group is a division of the Allen Institute. Unlike our other colleagues, we look externally. We look outside of the Institute to see where are the breakthroughs, where is the science that really needs that catalytic funding to push through. We have two primary mechanisms of support. One is the Allen Distinguished Investigator, and that's for an investigator or a team for over 3 years of funding at $1.5 million. Another mechanism is the Allen Discovery Center. These are larger teams and longer-term investments of up to 8 years and $20 million. Both investments are meant to hit high-risk, high-reward questions and really to move the field in directions that will have a lasting and sustainable impact.
There are very few places that are willing to really take this kind of risk, that are willing to say, we don't know exactly where this is going, but we believe in you. I can't think of a better investment in terms of payoffs. And history has shown that these investments pay off.
Most scientists are used to the idea and used to the concept that to make advances, one has to take risk. The real challenge is supporting those types of experiments, and most agencies are very conservative. The Frontiers Group is unique in the sense that they allow you to do experiments which at the outset don't have a defined end, and it's possible that they don't all work out. This ability to do these types of experiments is a real strength of the Frontiers Group, and it enables the type of thinking and the type of experiments that has the potential at the outset to possibly change the way we think about a problem, and perhaps even lead to treatments for diseases further down the line.
You have to be comfortable sort of with that unknown and with the fact that you might fail. It's not like a traditional funding mechanism where you can define in 5 years exactly what the outcome is. Honestly, if you can define in 5 years what the outcome is, maybe it's not that interesting or it doesn't push us in the same way.
For better or worse right now, science, it's very easy to get siloed. And this is why we hold multiple meetings a year, really trying to bring our researchers together to get new knowledge, new insights, and really cross-pollinate those ideas.
People from different disciplines, they look at things in quite different ways, often in very different ways. And so getting a different perspective can often nudge you in a direction that you hadn't previously thought of. And in the best cases, can even suggest a new experiment.
The best science or the real breakthroughs, they always occur at the edges between fields. And we really need men and women who can see the problem at the level of the atom, the cell, the tissue, the clinic, and bring that together to solve big problems. I would say the goal of our group is to make a difference at the end of the day, to find those ideas, to find those researchers that really have the possibility to move the field. They're out there, but right now the funding is such that it's challenging to find those high-risk, high-reward funders who will support that kind of exploratory work. And that's what we want to do. We want to make that difference and make an impact. Paul Allen's vision and also the Paul G. Allen Frontiers Group has made it all possible. You know, you can want to reach the frontiers, but unless you have individuals who are willing to bet on that, take risks on individuals and those ideas as well, it doesn't happen. So this is a partnership, and we go there together, and it's amazing to be part of such a visionary group.
Excellent. Well, I hope that video gives you a good overview of the Frontiers Group, and you're welcome to explore our website at allenfrontiersgroup.org and learn more. If you have any questions, please pop those into the Q&A panel, and my teammates Alexandra Basford and Casey Elkins will answer those for you. Now, turning to our program for the day, I'm very excited for this event. Our work at the Frontiers Group is always about anticipating the next great breakthrough, unearthing ideas that have potential to open whole new fields. Now, as you can see from the agenda, we've brought together a really diverse set of thought leaders, each with a different approach for studying brain health. And we've asked our speakers to imagine where the field is going and what their specific approach could teach us that might transform our understanding about human brain health and disease. Now, I invite you to be part of the conversation. If something resonates with you, call that out in the chat. If you have a question for the speakers, plug it into the Q&A panel, as Megan had instructed earlier. And before we start our program, we'd like to especially thank the late Allen Institute founder, Paul G. Allen, without whom this institute, this event, and much of our research wouldn't be possible. So without further ado, I'd like to introduce our first speaker of the day, and that's Marion Buckwalter. Now, Marion Buckwalter is a neurointensivist and a basic scientist whose lab focuses on how inflammation triggered by ischemic stroke affects long-term outcomes in brain health. And she is a professor at the Stanford School of Medicine and a deputy director of the Woodside Neuroscience Institute at Stanford. She also co-leads American Heart Association Allen Initiative team with Tony Weiss-Cray that aims to unlock the biological secrets of youth and rejuvenation in young blood. Now, her presentation is titled Vascular Health and Cognition, and we'll cover how cardiovascular risk factors, including high blood pressure, diabetes, and high cholesterol, are strong predictors of cognitive health. So without further ado, I welcome Marion Buckwalter.
Thank you so much, Kathy, for that introduction and for inviting me to speak today. It's an honor to be here to talk about vascular health and cognition. And the reason that I'm interested in vascular health is really because of the broadness of the problem of dementia and the severity of it. There's 56 million people alive today with dementia, and it's actually 1 in 10 people over 65. And not only is it a common disease, but it's really a dreaded disease. If you ask people, what disease do you most fear getting? At every age group, a plurality is that they say dementia. If people are over 65, 56% of them will say the thing they fear getting most is dementia. And that makes complete sense because obviously the brain is an important part of who we are and how we live our lives, basically. So I think because it's such a common problem, it's really important to approach it and in as innovative of a way as we can and as comprehensive of a way as we can. So I'm going to talk today for the first half of the talk about what we know about vascular health and why it's important for cognition. And then for the second half of the talk, I'll talk about where I think we need to go in the future to really solve this problem. So why blood vessels if you're thinking about dementia? This is going to be a common theme today, but since I'm the first speaker talking about blood vessels, I'd like to just spend a few minutes talking about how important they are. So the brain is a very hungry organ. It actually receives 20% of the oxygen and the nutrients that you take in and that your body uses, even though it's only 2% of body weight. And not only that, but the brain, if you want to rev up a part of your brain, say to listen to my talk, you actually need the blood vessels to dynamically deliver more blood flow to that part of the brain. So at a resting state, the brain doesn't have enough blood, it has enough blood flow to sit there, but not enough to be active and to work. So you have to have a dynamic vascular supply that is going to work to deliver blood flow directly where it's needed. And you can see here the major arteries wrap around the outside of the human brain. And then there are smaller arteries and arterioles that dive deep into the depths of the white matter of the brain. And these are the vessels that are unfortunately most affected by vascular risk factors. There are also some effects further in on the capillary, and I think you may hear about those from Dr. Gu later today. But vascular disease, in particular, aging and hypertension in this example, causes changes to these blood vessels that need to be dynamic and elastic. And instead of being dynamic and elastic, as they are in the young, they get narrowed, so the lumen that delivers the blood flow gets narrowed, and the wall gets less elastic and thicker and has more trouble dilating. There are probably also many other things that change about blood vessel function, but we have a lot to learn about exactly how this function works. And, And I use the example of aging and high blood pressure. They both do similar things, and so do obesity, diabetes, and high cholesterol. And I'd like you to appreciate that there's a complexity here. In humans, unlike in our animal models we use in research, a lot of times these things occur simultaneously. It's very unusual for one of these things to be seen without the others. Hypertension, for example, is very common, and in terms of dementia risk, it causes about 30% of dementia risk. And eating a high-fat diet, a sedentary lifestyle, genetic background, and time all contribute to these things. So they lead to vascular disease. And then vascular disease, presumably because of the problems delivering blood flow, leads to dementia. And you can see that here. This is a group of patients at UCSF, where you could see average memory is the dotted line here. And people with young, healthy, low vascular risk, so these people are not young, they're 65 and older, but they have low vascular risk, so their vessels are still young. Their memory stays stable over at least 7 years of this study. However, people with higher vascular risk, they start out worse in terms of memory, and they get worse faster. And I wanted to include this graph in my presentation because it represents a really important problem when we're thinking about treating dementia that's due to vascular causes. And that is that these changes in the vessels happen slowly over time. And what happens over here off the graph in midlife actually determines what happens later. And so if you're trying to prevent dementia in somebody who's already here, then our normal measures don't work. The second thing I wanted to bring up is that vascular disease itself not only directly damages the vessels, but it can lead to blood clots that can block blood flow and cause ischemia, or basically dead tissue inside the brain. That could be a large amount of dead tissue or a small amount. But just having that ischemia happen doubles somebody's risk of getting dementia over the 10 years afterwards. And we've recently discovered that this may be due to inflammation that's started by the ischemia and stays in the brain chronically. These are immune cells in the brain of a person that had a stroke and dementia. And this is a mouse model that we developed, which has similar, these are the same cells, just look a little smaller here, in a mouse brain. And in the mouse, we could show that they're actually causing the dementia after stroke. If we remove the cells, the mice don't get the dementia phenotype. So that's on top of the already misfunctioning vessels in humans as they age. So even just aging alone, but then on top of it, there's powerful influence from all of these cardiovascular risk factors. So what kind of treatments are already available for vascular dementia? Where's the state of the art in terms of the medicine of it? Unfortunately, there's not a lot we can do to treat it, but we do have prevention measures that work. And there's a lot of things you might read in the press, but actually there's pretty solid data that if you eat a Mediterranean diet, if you have high blood pressure and you get it treated, or diabetes and you get it treated, if you exercise, and if you stop smoking or never smoke, that it dramatically reduces your risk of dementia. There's a few other things that are not related directly to vascular risk factors that can prevent dementia, such as having more social activity, treating hearing loss, increasing the amount of education that people get, and treating depression. If you treat all of these risk factors for dementia, you can reduce the risk that people get dementia by about 30%. So that's pretty powerful, but when you think about how common dementia is, even with all of these things being perfect, there's still a lot of people that are going to get dementia. Some of it may be due to these early factors I was talking about, or factors we haven't yet discovered. So what can we do to help this problem? I think, here in the previous part of the talk, I just tried to emphasize that there's a lot of interacting factors. In many cases, they do end up affecting the blood vessels, and that's why our group is studying blood vessels, but there's certainly other effects as well. And then, as I mentioned, humans are not like mice. They're much more complex and have a long lifespan before they get dementia. That's affected, your diet is not the same throughout your life, nor is your exercise level. Your genetics is not the same as your neighbors, etc. So we need to embrace the complexity in order to go forward in the future. And as I mentioned, there's a big difference in size. So this is the size of a mouse brain next to a human brain. And I thought what I do to show you how I think this can work well is give an example from our work. But basically, I think we need to use animal models to understand pathways and to figure out if things are causative, basically, or are they just associations? What's causative? What's the molecular pathway? How do we develop drugs for that? But you could be studying something in a mouse and it might not be relevant to humans because of the major differences in lifespan and size, and in the case of blood vessels, the diameter of the vessels. So this is a case from Dr. Tony Weiss-Corray's research, who's co-leading our group with me. And he's been interested in aging for a long time, as Dr. Richmond said. And he found that this vascular molecule is increased in aging blood. And this molecule is shed into the blood with age. He found it in humans and also in mice. And it's something that's known to be involved in inflammation. And he was able to show that by blocking it in the blood, it blocks inflammation. So in collaboration with him, we went ahead, and this is work of my postdoc, Christy Zara, who's a fabulous scientist. We went ahead and blocked this molecule in old mice to see if it improved their memories. And in this particular test called the Barnes maze, a mouse is placed in the center of the circle, and they walk around, and basically all of these are holes that are open to the floor, but one of them has a little box the mouse can hide in. And the task is for the mouse to learn over several days how to find the box. So this is a typical graph of an adult mouse about 5 months old. And what you can see here is that on day 1, it doesn't really know where the box is. It takes a long time to find it. But by day 5, the amount of time it takes to find the box decreases. The mouse has learned where the box is, so it can go from the center to the box. However, if you do this with an old mouse, in this case a 16-month-old mouse, these aged mice, when treated with a control antibody, so this is like the placebo in a drug trial, they cannot find the hole as quickly. And this is something we all know about aging. We all get slower as we age, and this happens in mice too. So this old mouse takes much longer to find the hole. And then the question is, if we can block the substance that goes up in the blood, can we improve the memory of the old mouse? And when we did this, we were hoping it would improve the memory. We were really pleasantly surprised to find that actually here in blue, the old animal treated with the blocking antibody actually now has the memory of the young mouse. So this is something, I don't know about you guys, but I would like to have the memory that I had when I was 35. And that's about the comparative age of a 5-month-old mouse. So we're really excited about this, and we're continuing to study it in our animal models, and then also going back to humans. So this is an example of something where it seems to be in common between species, and we think that that's going to give us a much better shot of developing therapies that will help people. However, there's a lot of complexity out there, and the question is, how do you come up with these targets? And how do you find pathways that are changing that change during dementia or during old age or with vascular risk factors that are causative for dementia? And really what we need is we need to have new tools, and we need to refine the tools we have. So one of the things I think is really important is to take the complexity of biological information from people and be able to make sense of it and relate it to a clinically important phenotype. And this is just a graph of a different study I got from Dr. Agi Poor, who's one of our collaborators at Stanford. And here, what he's showing you in these gray lines and these many colored dots is multiple different data sets that contain like DNA sequencing or proteins in the blood or data about bacteria in the gut of people, and relating them to each other and saying which of these things go together. If they go together, they have a line in between them. And then what he does is he uses mathematics to make sense of this and say, well, which of these are involved in the phenotype you care about? In this case, cognition. And there's been a lot of advances in this in the last couple of years, and we're working on this for vascular dementia. We're getting plasma proteomics, so proteins in the blood from people, sequencing blood cells from people, and also relating it to cognitive tests and vascular risk factors like we discussed. And I think that this math is very good. I think as it gets better, it's going to really help us make sense of complex data sets. So I think that having somebody working on these topics who can bridge the gap between the biology and the math and help advance the math, as Nima is doing, really helps the research. The other thing that we really need as a tool is we need more information about the human brain. So the human brain that's been exposed to these vascular risk factors over a lifetime, and that's had different genetics from its neighbor, et cetera. And that we're here in the advent of single cell sequencing, where we can now take human brains and sequence them at a single cell level. However, it's been very difficult to get sequencing of the vascular cells. Most of that sequencing has focused on the neurons, which are obviously also important, but if the vasculature is the first thing to change and it's the cause of the neuronal dysfunction, then you want to know what's happening there. And so we now have the first atlas of human vasculature produced by an outstanding graduate student, Andrew Yang, in Tony's lab. And I think that sequencing human brains from volunteers who have had cognitive tests throughout their lifetime is going to be a good way to figure out what pathways in the brain match what's happening in the blood and allow us to develop treatments. And I think more tools like this combined with machine learning techniques to make sense of them is going to be really useful. So just a couple more thoughts before I wrap up. And I put this in actually because of a discussion I had with Dr. Richmond about tailoring science to one person. So the question is, do you tailor your experiments so that you get personalized medicine? You can say, okay, for you get this cocktail of drugs. And then for the other person, you test the non-green person next to them and they get a different cocktail of drugs. And I guess from my point of view, I think this is really exciting, but I worry that there might be issues with our healthcare delivery system where that means that we then have personalized medicine for the rich and nothing for other people. So I would advocate that we need to find a balance, but we should really think, as we develop treatments for dementia, we should really think about what can we develop that's going to be cheap, stable at room temperature so they can be distributed around the world and really help a lot of people. As I mentioned, cognitive aging is something that happens to all of us. And then dementia on top of it happens to at least 10% of us over age 65 and more as we get older. So it'd be great to think about that. And then Dr. Richmond also mentioned this, which is my final point, that multidisciplinary science and big team science really makes a difference. And I think that in this particular type of problem where you have a complex human disease and you really need to get down to the nitty-gritty of the molecular pathways that are causing the problems, you really need different kinds of experts to come together in order to solve those problems. In science, she mentioned I'm a neurointensivist, so I take care of people with strokes in the intensive care unit. And in stroke research, there's been a long history of basic science where treatments were found in rodents that didn't work in humans. But if you talk to a clinician about exactly the way the experiments were done in the animals, it wasn't practical. So for example, a treatment that you give to an animal 10 minutes after the stroke is never going to work in people because they're not in the hospital. We don't even know they've had a stroke at 10 minutes. So things like that are true all over medicine, and being able to just communicate about a disease, like this is the way it looks in people, how can we model that in animals? This is the way it looks in an animal, what people is that relevant to? I think is really important. And then I also mentioned having mathematics and analytic tools at our fingertips to embrace the complexity is really important. So I will stop there and take questions. Thank you very much for your attention, and glad to be here.
Wonderful presentation, Marian. I'm really excited to dig into some of the questions. We do have time for some of the questions that have been asked by the audience. And I'll start off with one from Jim Touchard. What is the role of neutrophil extracellular traps in vascular dementia?
Oh, great question. So for those of you who don't know, neutrophil extracellular traps are these really exciting things where neutrophils are these blood cells that are kind of like kamikaze warriors. When they come across infection, they spew out their DNA and they can make a web across a blood vessel. And that web can actually catch bacteria. So it's a design that works really well to trap bacteria and help the immune system kill bacteria that are in your blood. But when that happens, it can also clog blood vessels and impede blood flow. So it almost certainly is a pathophysiology that's involved when people have an infection in their blood and they have problems with their thinking. I think in vascular dementia itself, their role, as far as I know, is not well explored, but it would be very interesting to look at it.
Another question has come in from Simona. What is the mechanism for oops, it's scrolling off the screen. I'm sorry. Extravasation of white blood cells and ischemic stroke. So the liquid, and if you can define that for the folks who are watching from other fields.
Yeah, it's always hard. Like I tried to make it clear to people who are not scientists.
Yes.
There's several ways that immune cells get into the brain after a stroke. The first thing that happens is that all of the brain cells are really sensitive to loss of blood flow. And so the blood vessel wall cells themselves will break and immune cells can just get through them. There's also an immune response that's due to the dead tissue. So when brain cells die, they send out chemicals that attract white blood cells to that area. In terms of the long-lasting inflammation, we're still trying to learn why some people might have prolonged inflammation and others don't. But we think that it may be actually that there's a pro-inflammatory state from the vascular risk factors that cause them to get more revved up than they should. And instead of just coming in and clearing away the dead tissue, they develop aberrant responses where they're then damaging the brain. So I hope that answered the question.
Another question talks about superagers. Do they have better overall vascular health? What's known in that regard?
Yes, I think they do have overall better vascular health because the brain can't function without good vascular health. And I believe there have been some studies of superagers. So superagers are these people that don't lose cognition. And maybe you've met one of these people who are in their 90s and they're just as sharp as they ever were. So in a way, that's sort of the goal of the research I'm talking about, is to turn all of us into superagers so that we can stay ourselves as long as possible. And I don't study superagers myself, but I believe that people have looked at some of the arteries and other places in their body, and they definitely tend not to have diseases that lead to vessel damage. So my guess is that they would have young vessels to go along with their young thinking. But they're a really interesting population.
Well, there's more questions than there's time, but I have one last question that at least we can handle live from Sunil Apti. Blood vessels are not only cells, but have extracellular matrix as a crucial component. What's known about matrix remodeling in the context of the vascular basis of cognitive conditions?
Yeah, that's a great question. Remodeling of the extracellular matrix occurs in vascular dementia as it occurs in small vessel disease. So that the arterioles that I was talking about being most affected by vascular risk factors are also affected in hypertension and diabetes and other organs. And in the brain, like the other organs, there's this thing called mesenchymal transition, EMT, endothelial mesenchymal transition, where you get fibroblasts and they make different extracellular matrix molecules that make the walls of the vessels thicker. And that's one of the reasons they have trouble dilating. So it's definitely an area of interest. And one of the exciting things I think about the unbiased approaches I was discussing is that we will basically see what's there in the vessel. So we'll be able to see, and we'll see the immune cells that are there. So in terms of these questions, like we will be able to see, is it neutrophils? Is it extracellular matrix? And probably it's going to be many of these things. And then the question is, can we use mathematics to find common pathways that we can block to improve all of those things at once? Or do we need to look at multiple pathways? That kind of thing is on our minds for sure. So, and the other thing I just wanted to say, thanks for all the questions. I will have a look in the chat and try and answer some of them.
Yeah, there are definitely many more questions answered. It's really exciting to hear more about where the research is going and what's possible. So thank you again, Dr. Buckwalter, for participating today and being part of this session. And now our next speaker is Mukesh Jain. He is the Chief Academic Officer at the University Hospitals Health System, the Chief Scientific Officer at the University Hospitals Harrington Discovery Institute, the Vice Dean for UH Hospitals Affairs, and Professor of Medicine at Case Western Reserve University School of Medicine. Now, Mukesh leads another American Heart Association Allen Initiative team of investigators, and they are targeting to learn more about how red blood cells, the most abundant cells in our body, and the inner lining of small blood vessels called the endothelium work. His presentation today is titled Blood Vessels and Brain Health. Mukesh?
Thanks very much, Dr. Richmond, and good morning, afternoon, or evening, depending on where you are in the world listening, and thank you for joining us today. So I'm going to basically dovetail, or my talk dovetails well with Marion's beautiful talk on blood vessels and brain health. I'll begin with re-emphasizing a few points that Dr. Buckwalter made, tell you a little bit about what we're doing, and then looking ahead, talk to you a little bit about science in the future, but also the broader ecosystem that we need to develop to bring scientific discovery to its maximal potential. So with that preamble, I'll get started. And let's see if I can get this to work. Yeah, so Marion touched on this, and I'll just reemphasize. I'm part of the Allen AHA grant that's focused on dementia. And so I just highlight for you here something probably well known to most of the audience. Dementia is a big problem, an enormous source of morbidity, mortality, and healthcare expenditure worldwide, and it is going to increase over the ensuing decades. Now, I'm an adult cardiologist, and you might wonder where we became interested in brain health. And it really is because of something that Marion also emphasized, that there is really a very important connection between the heart and the brain. Both are energetically demanding organs, and they have common risk factors, be it age, genetics, hypertension, cholesterol, lifestyle. And I'll come back to this towards the end as well. The importance of the vascular part of the cardiovascular system is highlighted in this simple schematic of dementia, where you have common causes like Alzheimer's, but vascular dementia, either by itself or if it coexists with other forms, be it Alzheimer's, be it frontotemporal dementia, can significantly exacerbate disease. So understanding vascular health or the blood vascular system and its impact on the brain is very important. So the brain has a voracious requirement for energy. It's only 2% of the body mass, but consumes 20% of energy. Now, the business end of the vascular tree are called small blood vessels. Some of you may also know of them with the technical term capillaries. So these small blood vessels regulate blood flow and consequently energy delivery. It turns out the brain has very little in the way of energy stores. And so nature has wisely coupled the brain cells with blood vessels in an anatomic unit often referred to as the neurovascular unit. So you have neurons, astrocytes, and cells of the blood vessel, endothelium and pericytes in particular. Now, in our view, this diagram is missing a critical cellular element. And that cellular element is the red blood cell, which along with the endothelium, as I'll make the case to you, cooperate critically to regulate blood flow. So our group's sort of working hypothesis, if you will, or view, is that the most abundant cell in the body, the red blood cell, and the largest organ, the endothelium, together constitute a functional unit that controls blood flow, energy delivery, which is vital to maintaining cognition, brain health. And it follows that if you have loss and or dysfunction of red blood cells, endothelium, or both, be it with aging, be it with those risk factors that both Dr. Buckwalter and I have emphasized in the context of disease, that those would compromise blood flow, reduce energy delivery, impair the neurovascular unit, and impair brain health. So the biology that we've been studying, as Dr. Richmond emphasized, is focused on not the immune compartment of the hemovascular system that Marion talked about, but on the red blood cell and the endothelium. So the red blood cell, the most abundant cell, and the endothelium, the inner lining of blood vessels. And through a series of studies over the last couple of decades, members of our team have really advanced foundational biology, showing that red blood cells, in addition to carrying two gases, as we all learned in school, oxygen and carbon dioxide, actually carry additional gases. A form of nitric oxide called SNO, or S-nitrosylation, and indeed can also carry additional gases like hydrogen sulfide. And our working idea or model is that these gases are vitally important in helping blood vessels relax. They also augment in endothelial cells critical factors called KLFs or Krupa-like factors, and members of the NAD pathway like sirtuins that are really vitally important for endothelial cell health. And endothelial cells help relax blood vessels as well. So there's this positive mutually agonistic loop between the gases and these factors that controls blood flow and consequently energy delivery. On the basis of those discoveries, we're also working hard to develop therapeutic opportunities. And I will share with you in the ensuing slides efforts to leverage the insights in red blood cell carrying gases with a type of therapy called ENO, and then a second, a therapy called P7C3, which augments NAD and KLF levels in the endothelium. So the red blood cell. So the most abundant protein in red blood cells is hemoglobin. And hemoglobin on this diagram is represented as a dimer of 2 alpha and 2 beta chains. And in the mid-90s, our team member, Dr. Stamler, identified a cysteine residue that carries nitric oxide in a form called SNO. And this SNO, when released, allows the blood vessel to dilate and optimizes maximal delivery of oxygen and nutrients. The importance of this SNO is perhaps most beautifully demonstrated if you make an animal where you mutate that singular cysteine residue. And these mutant animals have reduced blood flow. They are susceptible to hypoxia. They have a compromised blood-brain barrier, shown for you in the bottom left, and that results in neurodegeneration. These studies have led Dr. Stamler and colleagues to develop a stable form of SNO called ENO, which you can give to patients. This is actually one of our team members, Dr. Reynolds, willing to be a participant in the science that he wishes to advance. And you can see that when he was made hypoxic at about 12%, not to worry, it wasn't too severe, but enough to cause his oxygen level and muscle to decline. And then we gave ENO at higher doses. You can see that we were able to recover oxygen saturation. The other therapeutic focuses on the health of the blood vessel and metabolic health of the neurovascular cells. Many of you in the audience may have heard of a cofactor that's really important in metabolism of all cells called NAD. Levels of NAD decline with age, and they are thought to be important. That decrement is thought to be important, not only the development of disease, but the very process of aging itself. Over the last 2 decades, my lab has identified a family of factors that also regulate aging and a number of the great diseases of today, at least in experimental systems. And they also regulate a number of the processes that are thought to be important in aging. So based on this, one of our team members, Dr. Pieper, developed an in vivo screen and identified a compound, has a funny name, P7C3, which augments NAD and KLF levels, has very high brain penetration, that becomes very critical, and it offers blood-brain barrier protection as well as neuroprotection. And I'll just share with you a couple of pieces of data on the next slide that underscore those comments. So on the top left is just one of many models where the Pieper lab has shown that after you do traumatic brain injury, you actually get breaks in the endothelial lining of little blood vessels. And even months or up to a year after that injury, if you give P7C3, a specific modified form that has particular high brain penetration called A20, you can restore endothelial integrity and improve cognition. More recently, they have advanced those efforts into the rhesus monkey model and shown that P7C3 increases neuronal survival. And his laboratory, and now many laboratories, have used the discovery of P7C3 to show that it is neuroprotective broadly across a broad spectrum of experimental disease models. And so the current state of efforts with P7C3 is that we are doing more rigorous studies in monkey, in non-human primates, with the hope that those studies can then be leveraged to advance to human studies after FDA approval. So that is some of the work that we're doing. And now one of the things Dr. Richmond had asked us all to do is sort of look ahead down the line, 5, 20 years in the future, and what we see on the horizon. And what I would emphasize to this group is that given the complexity of the diseases, the great diseases of today, of which brain diseases are one, cardiometabolic, cancer are others, we need to think in a much more holistic manner. And in cardiology, we've been doing this for some time. We think in cardiology a lot about our personal environment, genetics, habits, exercise, sleep, lifestyle, microbiome. We think about the social environment, the impact of pollution that is well documented, the impact of the natural environment, day-night cycles, which remarkably in cardiology, day-night cycles are linked to sudden cardiac death or stroke, heart attack, and exacerbations of heart failure. Now, given the linkage between cardiovascular and brain health, I think it is reasonable to think about the neurovascular unit and brain health moving forward in a much more holistic manner, to study the impact of personal, social, and the natural environment, its impact on the neurovascular unit. How do the cells of the neurovascular unit respond to these inputs and consequently impact the organ broadly? Are there convergence points? Are there conversion points between pollution, some of the personal environmental cues, and day-night cycles, for example? Identification of those convergent points might be very important and can be leveraged for new and emerging therapeutics, be it gene therapy, novel biologics, nucleic acid therapy, and top of mind today, genome editing. And my guess would be is that 5, 10, 20 years from now, the way we're really going to combat the diseases of today, like dementia, which are so complex, is really through a multi-pronged approach. Now, at the outset, I noted for you, I'll tell you a little bit about what I see in science, and that is what I've just covered for you in the last couple of minutes, but also some comments that grew out of a discussion with Dr. Richmond about the broader ecosystem. Because the truth of the matter is, and you're going to hear great science today, but the truth of the matter is that we need to really align that science with the broader ecosystem to achieve the full potential of our discoveries. And failure to do so will leave many of these discoveries on, as we like to say, on the laboratory shelf. So the diagram on this slide, I think, serves as a good launching point for my last set of comments. This is a diagram that in different iterations is probably well known to the audience. We often think about basic translational, clinical trials, population health, as all being important in a continuum to impact patient, community, and world. There are challenges with each of these steps, and I'll start from the right and work backwards. Population research, the adoption of practice guidelines by patients and physicians, for that matter, can take much longer than you would expect, often over a decade. The regulatory challenges and cost of large randomized clinical trials. And perhaps in an area that's most acutely of interest to the audience today, the challenge in basic and translational research, the movement of discovery from the laboratory to the point where it can be commercialized. And what we need is not only the discovery, but we need partnerships across multiple institutions. We need, as Dr. Buckwalter also mentioned, team science, interdisciplinary research to bring or to advance the most impactful therapies to combat disease. And if you don't believe me, you might believe someone who's a lot smarter. This is Francis Collins, quote, a very nice quote from Francis from a few years ago, obviously our NIH director, in reference to this diagram, the reduction in drug approvals in the US, FDA drug approvals. And he notes for you that we've had a great outpouring of discoveries, and we will continue to do so with support from diverse constituents, the government, foundations, amongst others. Yet paradoxically, our ability to turn that information to clinical fruition has slowed. And if there was ever a time that we need to figure out how to take advantage of the great science that is being done and will continue to be done, and apply bold, audacious principles of innovation to make products that impact human health, now is the time to do it. What Dr. Collins is referring to is a metaphorical chasm that many of you may have heard of in basic and translational research called the Valley of Death. That gap between research in the laboratory and the ability to commercialize that research to make a medicine. And we need bridges. We desperately need bridges to traverse the valley. There are efforts underway to do this, and I will share with you in the last couple of minutes an effort in our community to try to traverse the valley called the Harrington Project. And again, there are additional efforts being done in the state of New York, the Parker Institute, and others that are trying to do it in different domains. But let me tell you what we have tried to do. So the diagram here, if I draw your attention, really at a high level gives you the model of the Harrington Project, which is that we have tried to align in academia and developed a unique nonprofit for-profit model that has three major components. We select and source the best science across the nation and now across the United Kingdom, and we're guided by a spectacular scientific board. Once we identify that science, we surround that investigator with a therapeutics development team, 24 individuals that have sector expertise across these domains, literally giving pharma level capability in academia. And we help de-risk those high risk, high potential discoveries to the point where a for-profit partner might wish to advance that discovery to a medicine. Over the past 8 years since inception, this is our footprint. We have supported investigators across North America and now the UK. And of the approximately 120 so projects, we now have 5, and I believe we'll have a 6th very shortly, technologies that have been licensed to pharma. So looking ahead, along with the great science that you will hear about and will be proposed, I will just underscore the importance of having the entire ecosystem evolve at the same time. And we have to evolve and optimize coordination between these domains, these institutions, which will be vital to achieving the full potential of what we seek to achieve in addressing unmet medical needs. And with that, I just want to thank the audience again, thank Dr. Richmond and the Allen Frontiers group. This is a picture of our team that is addressing the blood and endothelium that I've covered for you today, and some of our scientific advisory board members that help us think through the challenges that we're trying to tackle. So with that, I'll stop and turn it back over to Dr. Richmond.
Thank you so much for that wonderful presentation. I'd be remiss if I didn't call all the participants to say, let's give a virtual round of appreciation to our first two speakers. It's a wonderful way to start off these discussions, and we'll be adding to the chat actually more information about both of their team's work from the team from Stanford, co-led by Dr. Weiss-Cray and Dr. Buckwalter. We've highlighted some of the work that looks at how old age changes the way blood accesses the brain. And then from Dr. James' team, we're also going to touch on something he didn't mention, is how some of this research is actually being pivoted to actually help patients suffering from COVID. So feel free to find those links in the chat. And I think we have time for one question, and there are many questions. So I will go to one that I think you may already have the answer to, because we don't have much time. And that's the one actually that was also asked of Dr. Buckwalter, the role of these neutrophil traps in disease, and what your thoughts are on the role they may be playing, particularly given the angle that you're coming at.
So I think neutrophil traps from our perspective impact the blood-brain barrier. And there is pretty good data, some of it that we have within our own group, that neutrophil traps impact endothelial health, endothelial activation, and that would result in leak of the endothelium, and activation would recruit those immune cells from the blood into the brain, which can increase damage. The nets are also interesting because they impact the cardiovascular system, where they've been studied pretty strongly. And in fact, if you block net formation, you can alleviate, for example, heart failure consequent to ischemic injury. So maybe something like that, be it in large vessels, stroke, or small vessels that can result in dementia, maybe that approach would be therapeutically tractable in brain disease.
Well, I know that you have a lot of questions waiting for you in the Q&A. So thank you again, Dr. Jane. Wonderful to hear your thoughts, and definitely not only where science can go, but maybe how we do science can help improve some of our impact. Our third speaker, welcome Dr. Rusty Gage. Dr. Gage is the president of the Salk Institute, professor in the Laboratory of Genetics, and holds the V. and John Adler Chair for Research on Age-Related Neurodegenerative Disease. He also is leading another AHA Allen Initiative team. It's an interdisciplinary group of professors at the Salk Institute that believe that Alzheimer's disease and other age-related brain disorders are triggered not by a single event, but by a failure of complex interwoven biological systems in our body that start to break down as we age. So his presentation is titled Future Directions for Science, and he'll be also talking about somatic mosaicism. So with no more further ado, welcome Rusty.
Thanks, Kathy. It's a pleasure to be here and join the other speakers in this really important Frontiers presentation. So first of all, thanks to the other speakers for their very clear and lucid presentations. So our project really is based on the need to understand the cell biology that drives aging, and with the firm belief that age is the major risk factor in neurodegenerative diseases and cognitive impairments that occur with age. And this is really a collaboration with 10 of us, all professors at the Salk Institute, that approach this problem of aging and dementia from different perspectives. And we've pooled forces to work together in a common way. So let's see here. So traditionally, how people have thought about aging and Alzheimer's disease is to pick one of these topics and drill down into it. Say it's energy deficiency that leads to dementia, or inflammation, or some epigenetic change that occurred early on, or more than likely some sort of genomic integrity where the DNA of individual cells begins to decay. And what we're beginning to find is that these are not independent processes, that they work in a state of homeostasis. That means that they work together, supporting each other. So energy metabolism defects often lead to inflammation and subsequently into genomic integrity. So our goal is to monitor all of these systems simultaneously as we age and as this leads to dementia. Now, our approach is at multiple levels, and we look at individual cells, and so we have a focus on neurons, glial cells, endothelial cells, and not as individual cells, but how they cooperate with each other and how they change with time. We also use aggregates of cells in an organ called an organoid, which allows for these cells to integrate, and they're from cells that are derived from human skin and then reprogrammed into a variety of cell tissues that can self-aggregate into a microstructure that has similar characteristics of brain tissue. And then finally, we have an aging marmoset population with that show an age-related cognitive decline, and we're monitoring those with therapies that we generate from our individual experiments. So I'll give you a little bit of an example on a couple of these, the progress that we're making using these different levels. And I'll focus primarily on the individual cells as well as on the organoids and the progress we're making with them. So a discovery we made some time ago has really opened up this area of aging for us. You can take from any individual a biopsy, a skin biopsy, grow your skin in a dish, and then overexpress a subset of cells, of genes in those cells, and they can be forced to differentiate the cells into a mature neuron, which has the genetic makeup of that individual. Interestingly, if you take another approach, which is to take the fibroblast and reprogram those cells into stem cells, then you erase all the aging program that was in that fibroblast and treat it like it reverses back into an immature cell. But that's of the same individual. And what this has shown us is that age, or if you can retain the aging program within the cell, you can capture many of the phenotypes that are associated with that individual. So an example of that is here. Here's a cohort of 15 individual patients diagnosed with Alzheimer's disease and 15 controls. And we compare their RNA expression patterns in the derived neurons, one from another, to determine whether or not we can find any Alzheimer's specific age-dependent differences between the cells. And a clear signature begins to emerge. And that is that while there is a change in energy metabolism, there is a remarkable shift in the function of this energy metabolism from its normal use of oxidative phosphorylation into glycolysis. This is interesting because this is what's happening here is the cell, a mature cell, which normally is using oxygen to provide energy, is shifting back into a state where it's using glucose. That's very reminiscent of what happens with cancer cells when they begin to proliferate. And in our case, we're seeing that in Alzheimer's disease, the cells are beginning to move into a de-differentiated state where they appear to seem as though they are trying to undergo proliferation. Another finding that amplifies on this is that when we look at the chromatin of each of these individual cells and examine what spots of the chromatin are open, we find that those spaces in the DNA that are open for activity really reflect what we're seeing with this de-differentiation of the cells. We can see that the cells have lost their neural connectivity functions. Genes that are activated are involved in cell death, and also genes that are involved in cell cycle reentry, this de-differentiation of the cells. So the schematic architecture of the genome itself in these individual cells from these individual patients represents this lack of differentiation in Alzheimer's disease. The third thing that is quite apparent is that there's a lack of ability of the cells to repair their own damage. So genes that are normally involved in repairing damage when it occurs in a cell have gone, are decreased dramatically, and the whole system of DNA repair. Normally, there's about 400 genes in the DNA that repair there are 400 proteins in the genome that repair DNA as it's damaged with age, but those are downregulated in Alzheimer's disease specifically. And there's an upregulation of molecules that are involved in inducing damage. These are just some of the, overall, we see that there's a genomic instability in Alzheimer's disease, independent of age. So you should remember, in all cases here, we're comparing age-matched individuals to AD. So this is an AD-specific feature. We've developed a tool, and I'm not going to go into too much detail, but you can imagine now that what we're asking is, where is this DNA damage occurring? Are there areas of the brain, of the genome that are specifically susceptible to damage, or is it random within the genome? And this tool that we're calling a RepairSeq is a mechanism to identify exactly in each individual cell where DNA repair is occurring and where DNA repair is lost with age. So in the last couple slides here, I'm going to show you some examples of advances that we're making in these organoids. One of the great tools that I've just been telling you about is how you can take individual cells from people, convert them into really any cell of the body, and examine their features. But more recently, we've been able to take those, and many others have taken cells from that patient and allowed them to self-aggregate into what is called an organoid, and then examine here, instead of individual cells, a more complex organ. One of the downsides of this, however, is they're not vascularized, so they don't have all the features that you just heard about. They're mostly neuronal. So we've been working to develop a system where these organoids can be vascularized and also contain some of these other important cells we know about, like endothelial cells, microglia, and astrocytes. And just as a feature, we've been able to take these human organoids and implant them into an immunocompromised mouse. And what happens is the vasculature grows into the organoid, and they survive indefinitely, as long as the animal survives. And we've been able to do this and enrich the cells with human microglia. So here you see an organoid inside the brain, and these are human microglia that are patterned within that tissue. In the normal in vitro setting, we really can't get the microglia to survive, but clearly in this organ we can. Another cell that's been missing from the organoid cluster are astrocytes, which I think you've heard a little bit about. This is a normal organoid with bright neuronal features to them, but here we have these glial features. If we zoom down into the cells and label the individual cells in situ, we can see these astrocytes in their full glory. Now what's remarkable to us is these are human astrocytes, and they really have very different properties from mouse astrocytes. So this is really the first time we'll be able to examine in vivo human astrocytes. One of the good features of this is that we can also now examine human response to inflammation. So here we have the organoids treated with IL-1 beta or TNF alpha, which are cytokines that are generally involved in inflammation. And you can see that they're clearly an upregulation of these cytokines like IL-8, which is a responder cytokine. And if we look very carefully, we can see that the glial cells in here are just flooded with IL-8. So we have now a system to monitor the normal and activated state of inflammation. So I've talked to you a little bit about several of the systems that we're particularly interested in: inflammation, DNA damage, mitochondrial function, and vascular integrity. But I want to focus in this idea that Kathy challenged us with in thinking about the future of brain health and where are the gaps. And this is one of the gaps that I feel is crucial for us to understand. It's sort of the dark matter of neurodegenerative systems. That is, how can we learn more about genomic integrity? We believe, and we're growing evidence that there is an age-related decline in the vascular system with time. Carl Harrop was one of the first to really highlight this in this wonderful diagram showing that normally with age, if you look down at the bottom of here, which is DNA capacity, so there's a normal increase in DNA damage that occurs with time, and then it sort of reaches a threshold where any intervention or perturbation takes it on a drastic decline. And this is where we think of normal aging going this way, and some block or interruption of that homeostatic mechanism that happens, and then this decline occurs. So just to remind you, DNA repair is crucial, as I said earlier, and there are a number of ways in which DNA lesions can occur and a number of wonderful systems for repairing it. However, post-mitotic cells, like neurons, selectively downregulate many of the DNA repair pathways because they're not dividing. It feels that they don't need to put their energy into making those repair systems. So they are downregulated. Nevertheless, quite a fantastic number here. Every cell of the body, even post-mitotic cells, undergo more than 1,000 DNA damaging events every single day. So that gives you a feeling for how active the repair system has to be. And when DNA damage is disrupted, which it often is, in particular in long genes, and as we all know, or we've learned, neural genes that are expressed when neurons are functioning, are often very long, and they're very susceptible to DNA damage. And what we're showing, and what we've shown in others as well, is that aging and neurodegeneration is linked to an increase in the mutation in these DNA damaging molecules. So a little bit further. So post-mitotic cells inactivate many of their DNA repair, as I said, and these deficiencies in these remaining pathways lead to, in many cases, a cell-specific and phenotypic specific functional outcome. One example is in progeria, typically associated with neurodegenerative diseases. And these less penetrant alleles are likely to be manifest in age-related degeneration. This just shows this lack of the downregulation of many of these repair processes that occur in post-mitotic cells, whereas in replicative cells, they have more ways to repair. This lack of reparation here results in a cellular aging is occurring more rapidly in brain cells, which are post-mitotic. So just to remember now, these events that I'm talking about are post-mitotic. So you and I, all of us, do not have a single genome within our body. Each of our cells are slightly different from each other in that they may have a large copy number variation, or they may have a point deletion. And these events occur early in development in some cases, or they can occur late in development. So if the event occurs, this is parents, this is the offspring, and this is the cells, the initial zygote. And if a mutation occurs there, then the mutation will be seen in all the cells. And you can pick this up very easily using bulk analyses. However, since these genomic integrity events occur throughout life, if it occurs very late in life, only a few of the cells may be affected. And this is, I think, the bottleneck. This is the area that I'd like to talk to you about. We don't know how DNA repair might allow cells to somatically evolve. So if you have an event that occurs early in life, it may not have much effect on your body until you're old, when this homeostasis is beginning to decay, then that early event may show up as an error. Does the body and does the genome have a mechanism for targeting the repair of certain mutations that occur early that may not have a consequence until subsequently? We have no idea of the strategy that the body uses to repair itself. We don't understand if there are distinct differences in the areas of the genome that are damaged, and if this differs among neuronal types or different between different cell types. So this is my challenge for us. There are unrepaired mutation events that occur in the brain during development. and they can have big effects later on in life or even early on in life. But these changes in DNA occur throughout life, and this results in an emerging concept that we have, that we are a walking somatic mosaicism. We have different genetic profiles in each of our individual cells. Current genome sequencing technology struggles to sample enough cells with deep enough coverage at an affordable cost to capture the accurate representation of your genomic complexity. Right now, we're treating it as if you have a single genome, when in fact you're living with multiple genomes in your body. In addition, this unknown mosaicism within each individual, we know little of the effect of age, of gender, ethnicity that drives the individual diversity. So we can't just talk about this as individuals and our own. We need to look at systems that are different in different genders, in different ethnicities, and how those different features of an individual and a population affect their ability to repair. Do we have different mechanisms for how we repair and which genes we repair or during a lifetime? In the future, and this is a challenge, as is most often the case in science, where technology will drive things. We need rapid, inexpensive, and high-resolution methods and instrumentation to determine how somatic mosaicism provides a unique background on which disease of the brain is manifest across age, across gender, and across ethnicity. So with that, that's the challenge that I think we face in the future, and not treating ourselves as a common genome, but really looking at somatic mosaicism as a background on which disease flourishes. So these are my colleagues, and we're challenged by looking at aging and its consequences on cognitive decline, and we are trying to integrate different systems to examine how individuals decline and with age. Thanks.
Wonderful. Thank you so much, Rusty, for that exciting presentation. We have a number of questions, and we're running a little bit over, so I'm just going to pull one, but the rest can be answered as well offline. You don't often hear of somatic mosaicism. To what extent is this captured or seen in brain organoids, and to what degree should it be accounted for in experimental design?
Yeah, so somatic mosaicism is profoundly seen in the nervous system. So neurons, glial cells have this, and you can definitely capture this, and we are capturing this in organoids, but even in single cells. You can take normal monolayers of human neurons and label them with a marker that indicates exactly where this DNA damaging event occurs. And that's why we developed this repair-seq system, so that we can mark every spot in the genome where a repair event occurs across individuals and across time. So yes, it does occur in individual cells. You can monitor it in organisms, and you can look in postmortem tissue and of course see where all that DNA damaging event occurs. I might add, just as a side point, because that's such a good question. If you look at the damaging events that you can detect in a postmortem person, and you're not finding where the repair sites occurred, you're just looking at it with damaging occurs, those spots where the damaging events are observable in postmortem tissue are different from the sites where repair is occurring. It's almost as if you're allowing those areas to be detected. We need to find out where the repair sites are and compare them to the detectable changes.
Excellent. Well, thank you so much. That was very exciting. It was great to hear from you. And on to our last speaker of the morning. It's my pleasure to introduce Chenghua Gu. Dr. Gu is a professor of neurobiology at Harvard Medical School and is a 2018 Allen Distinguished Investigator. She's expanding our understanding of the brain's energy and environmental control, since it's critical for promoting optimal brain function and treating brain disorders. Her presentation today is entitled "Our Brain's Control of Its Own Energy and Environment." And without further ado, welcome, Chenghua.
Great. Great to be here. Thanks, Kathy. And also I want to thank all the previous speakers already introduced you very nicely. Our brains are heavily vascularized, and it's highly metabolic demand, high energy demand, right? This everyone mentioned is 2% mass and 20% energy. So I don't need to convince you more. But I do want to mention that neurons in the brain are extraordinarily sensitive to their environment. So they have both high demand of energy, also very sensitive to the environment. So luckily though, our brain has evolved to have the capability actually control its own energy and environment. So to meet the moment-to-moment changes of energy demand, we have this neurovascular coupling mechanism. So basically neuroactivation increase local blood flow very rapidly. And secondly, the brain's chemical milieu must be tightly controlled, and this environment is controlled by the blood-brain barrier. So my lab is in general interested in understanding the basic mechanism underlying both physiology of the brain. So today I'm going to share with you one example of each to demonstrating how the energy supplies is regulated by neurovascular coupling, and then how the environment is controlled by the blood-brain barrier. And then I'm going to show you my vision how we further understand this in the next decade. Okay, so first about the environment. Blood-brain barrier maintains the homeostasis of the brain environment. Basically, it's a gatekeeper of the brain, control things go in and out, also protect the brain from the toxins and pathogens with very limited entry of the immune cells. And here's just a cartoon version, but I think by now everyone knows blood-brain barrier resides in the endothelial cells, form this single layer of blood vessel. All the green stuff are the axons, dendrite, synapses. And here's the astrocyte with the end feet hugging the blood vessels. So have a tightly controlled blood-brain barrier, no doubt, is very important for the brain function. But at the same time, it's also the biggest obstacle for the deliver drugs to the CNS, treat any of the neurological diseases. So in this case, we would like to be able to transiently open the barrier to treat all those neurological diseases. And also on the other side, the coin, recent research also demonstrate that the barrier breakdown precedes the neurodegeneration. So in this case, perhaps we wish we could tighten the barrier to delay the disease progression. So understanding the basic mechanism will enable us to dial this, manipulate the barrier in both directions for disease and repair. Okay, so why the blood-brain barrier is impermeable? So now I'm open up the tube, the blood vessel. So one side will be the lumen, and the other side will bring parenchyma. So here's the three adjacent endothelial cells form the wall of the blood vessel. So normally you can imagine things across from one side to the other, either go between the cells or go through the cells. And the blood-brain barrier, the impermeable nature historically largely credited to the specialized tight junctions to prevent things go between the cells. But you can imagine, like many other cell types, the substance also can take upon by the vesicles and through a transcytotic mechanism goes to the other side. So we discovered that this mechanism, transcytosis, is actively inhibited at the blood-brain barrier. So together with both mechanisms, ensure the integrity of the barrier. So here's some evidence. So we first identify candidate genes, which means they're enriched in the brain endothelial cells, but very low expression in the lung endothelial cells, which doesn't have the barrier. So for the past few years, we've been concentrating on studying this one multi-transmembrane protein, MFC2A. So here, when we inject, it's a red fluorescence dye through the tail vein of a wild-type mouse. The typical you're going to see, they're completely confined in the blood vessels. However, in the MFC2A mutant mice, the tracer leak out of the blood vessels, stain the brain parenchyma. But to our surprise, MFC2A knockout does not affect the tight junctions. So here we inject the electron-dense tracer to the tail vein of the mouse. Look at this under EM, and you can see both in the wild type and the mutant cases, the tracer stop very sharply at the junction, indicating the junction is functionally normal. However, the knockout mice this inhibits transcytosis. So in the wild type case, you don't see any traces in the endothelial cell. However, in the mutant mice, you see the tracer containing vesicles in the endothelial cells, indicating that leakage is not due to opening of junctions, but rather upregulation of the transcytotic vesicles. So normally, MFC2A must inhibit transcytosis. And what type of vesicles are those? To make a long story short, we identified is the caveoli vesicles that are particularly inhibited at the BBB. So caveolin-1 protein is obligated component for caveoli vesicle. Without this protein, those vesicles will not form. So here shows immuno-EM of caveoli-1. Those are the caveoli-1 decorated caveoli vesicles in the mutant mice. So we demonstrated that MFC2A suppress transcytosis by blocking caveoli formation. Just remember the MFC2A mutant mice have increased vesicles here highlighted for you. And in the double mutant mice, MFC2A and CAV1 mutant mice, this phenotype is completely rescued. And mirror this result, the leakage also completely rescued in the double mutant mice. So that means the upregulated caveoli vesicles account for this leakage phenotype. Again, normal function for MFC2A is to inhibit this caveoli, specifically caveoli formation. So how does this happen? Well, turn out to be MFC2A is a lipid translocator. So it's flip the lipid from, especially the unsaturated phospholipid like DHA, from the outer membrane to the inner membrane. As a result, it's changed the lipid composition of the plasma membrane, and this lipid composition inhibit the caveoli formation. So this is one of the first mechanisms demonstrating how our brain endothelial cells actively inhibit transcytosis. We're particularly excited about this mechanism because seems like unlike the tight junctions, which more static, at least from what we know, right? So this mechanism seems more dynamic, and indeed it turned out to be true. We recently published that both during development and also during disease, indeed this pathway is very dynamically regulated. So this opened a huge opportunity for the drug delivery and therapeutics, because we want to harness our discoveries, able to dial backward and forward to open and close the barrier for the CNS drug delivery and also repair. So what I told you is obviously just the beginning of understanding the barrier. So currently, we continue to identify other key molecules other than MFC2A in the endothelial cells, and we also try to understand the surrounding parasite and astrocyte, what their signals to the endothelial cells maintain their integrity. And also, this actually thanks for Alan Frontier's support, we also start to address the blood-brain barrier role, which actually anatomically sitting right before the periphery and the brain parenchyma. Its role, how do brain endothelial cells can transmit the periphery immune signals to the brain. And then obviously we want to harness the discovery for the drug delivery and treatment of neurodegenerative diseases. Okay, so I'm just going to now switch to the second topic, the energy control, to show you one example. The energy supply is regulated by neurovascular coupling. So during behavior, various brain regions circuitry are brought online to mediate those behavior events, and the resulting neuroactivity rapidly increase the local blood flow. So this is called neurovascular coupling. This is obviously very important to day-to-day life of the brain to meet moment-to-moment changes in the regional brain energy demand. And turn out to be this dynamic functional coupling is also the basis for the functional brain imaging in humans. And more recently, emerging evidence show that, and many speakers already mentioned, its impairment contribute to the neurodegeneration. Despite the importance, the underlying mechanism still poorly understood, partially because this level of imaging is not enough to provide the temporal spatial resolution needed for understand the mechanism. So my lab has been doing is to use imaging tools to study in awake mice with natural physiology stimuli and at a single vessel resolution. So here I'm just going to show you a movie. You can see this mice running on the ball, and then we have this 2 photon on top of the corneal window looking at the barrel cortex. So you can see as the foam stimulating the whisker pad, here you see the quickly increase of a neuroactivity indicated by GCAM in neurons. And here's one blood vessels, arterial blood vessels labeled, and you can see the vasodilation and then return to normal. So in addition, we also have the capability to look at the capillary blood flows. Together with Mosaicism, we are currently investigating new models of the neurovascular coupling. The previous speakers already introduced you very nicely the brain vasculature network. So the neurovascular coupling started with the increased neuroactivity and ended on the smooth muscle cells, which wrap around the large arterial blood vessels. The whole process happened in vivo in awake animals is less than hundreds milliseconds. It's really fast. So traditionally, the field thinks this happens simply because neurons can send vasodilatory cues directly actually on the smooth muscle cells. However, many neurons are not conveniently nearby here. So we and others begin to explore a second mechanism, which we think the neurons' increased neuroactivity can sense by the capillary endothelial cells, which then rapidly transmitted via the vascular tree and then actively tell the smooth muscle cells. So just to make a long story short, we recently just published a paper demonstrating that when you perturb the signal, particularly in the endothelial cells, arterial endothelial cells, indeed you can change the outcome of the neurovascular coupling. So I'm just going to show you one piece of the data from that paper. So this is in a stark contrast to the previous movie I showed you, which has happened in the wild type mice. So here is a mouse, particularly knockout one gene in the arterial endothelial cells, and you can see following the whisker stimulation, although the neuroactivity increased, but you don't see the vasodilation anymore. And in addition, I'm also going to show you the, so here is a camograph of the capillary blood flow. So again, in the control mice, you can clearly see by your eye the rapid increase of the blood flow. And this is also not happening in the mutant mice. So currently, we are continue to investigating, how the signals are rapidly propagated through the endothelial cells to the smooth muscle cells. And equally important, we also try to understand how the signals are being stopped so that the energy can be used very economically instead of it's very locally dilation instead of the entire brain just dilated. And then finally, we also now have genetic tools that allow us to address the consequence of impaired neurovascular coupling for brain function. It's aged earlier or it's actually developed a neurodegeneration. So we're really excited about those lines of work. What I showed you, obviously just a tip of the iceberg. It's a beginning to address how our brain control its own energy and environment. It's no doubt the environment energy is super important for the well-being of the brain. So what what should happen and can be happen in the next decade, how to in this area of research. So again, I'm going to divide it into two parts. One is about the environment, and one is about energy control. So first is about the environment. So I want to say that the blood-brain barrier, what I just told you, is only a piece of the puzzle of the entire, I call brain fluid dynamics. Don't forget our brain entirely based in the cerebrospinal fluid, right, CSF. And in addition, this has to drain out through the lymphatic vessels on the surface of the brain. So really, it's the brain dynamic is all the different roots constantly moving, interconnected to each other, and they are the ones control the nutrient delivery, waste clearance, and signaling. So at any given moment, when we talk about the brain environment, your actual neuron you care about, exposed to the environment, is a combination of all those roots, all those different processes. It's a balance of those, right? So however, I'm simplifying this cartoon at different pieces, but the truth is, even right now, the biological substance substrate underlying each of the root is not even clear, the biological structure, and also the directionality is not clear. For example, people often take brain interstitial fluid, ISF, equivalent to CSF. But the truth is, many of the waste is being actually transported back to the circulation via the blood-brain barrier. So what proportion go to this pathway, what proportion go to this pathway is not known at all. But often this is used as a biomarker, but they're not equivalent. In addition, we know the CSF eventually will be drained out to the lymphatic. On daily basis, a human being is like 100 liters of that, right? But what is the biological structure underlying this is not known. So we're facing a huge challenge, right? So we need to first characterize the anatomical connections between the brain fluids. We need to measure in the fluid dynamic in the living brain, because many experimental manipulations confound the result. Anesthetized versus we get totally different result. When you inject tracers into the CSF, you're adding additional pressure, also perturb the system. So one way to solve this, as every speaker's already mentioned, always the new technology development. So in this case, particularly important, we have in vivo system. We need to leverage the emerging imaging platform and develop new method. So I don't think I need to convince you the impact. If you can understand more about the energy and environment, no doubt you're going to provide new insight into neurodegeneration. And then from a discovery point of view, also is review new target and also very important biomarkers, right, in all those fluid. And also identify the potential CNS drug delivery routes. So the final point is about the energy. Again, I just touched the tip of the iceberg, showing you the energy supply by neurovascular coupling. So how the brain actually utilize those energy, right? It's surprisingly very little known about it. So I call the cellular fuel utilization metabolism. So for that, I decided to actually show you as a vignette, a very elegant work from my colleague Gary Yellen's lab, who's in my department. So to particularly point out that even the source of the fuel you use is completely make a huge impact on the accessibility of your brain health. So the things that inspired Gary's work was this really interesting, very effective, low carbohydrate, high fat ketone diet for treat epilepsy patient. So Gary wanted to know at a cellular molecular level, how this happen, why this work. So in order to do this, he has to first develop a set of tools. So for example, his lab developed the metabolic biosensors, allow individual neuron in real life track the metabolic signal. Here you can see from the movie. In addition, recently he collaborated with Netherlands lab using mass spectrometry imaging of metabolism. So we all know mass spec can capture information of thousands of metabolites at the same time, right? But then this technology allows you map back to its natural location on the brain slices. So this is incredibly powerful. The combination of those can give you a temporal spatial resolution, a cell type specific manner. In addition, Gary also can see the downstream neuronal function by electrophysiology, calcium imaging, EEG. So it's really the combination of all those technology together finally allowed his lab discover how this magic diet can make those patients become resistant to epilepsy. So I hope this example just give you a sense that even the one particular source of the field, you need so much work, and there's so much more we need to know. So the challenge is we need to understand the field sources, the pathways, and the regulations. And we need to know this at a cell type specific temporal and spatial resolution in the native tissues. And we need to simultaneously monitor thousands of metabolites. And again, the bottleneck obviously is a technology development. We need more fluorescent sensors. He made a few of them, so we obviously need more of them. And this mass spec imaging, we wanted to have the capability to have a temporal resolution, and also develop more toolkits to track a different pathways. So again, the impact I think is very, very obvious. So the to first understand that this, I think from basic biology point of view, is still a gap of knowledge. And also, I think there's also emergent evidence metabolism is altered in neurodegeneration, and may even possibly play a causal role. So I think from therapeutic, this is actually very unique, because besides you have new biomarkers, a dietary manipulation probably safer and more cheaper than drug delivery. So I think this probably have a huge impact on that. And of course, a potential drug target. So here's my lab. And then I particularly want to thank Paul Allen Frontier to support the work. Thank you.
Excellent. Thank you so much, Cheng-Hua. It's exciting to see those latest results, and how elegant and how dramatic they can be. We do have, we're running a little late, but there's one question. There's many questions, but there's one question I think we have time for. To what extent could gains be had by viewing this with a physics or engineering lens when it comes to fluid dynamics? And what are your thoughts regarding blood-brain barrier on a chip methodologies?
I think it's a really interesting question, because I think many people try to do that, and then it will be wonderful if this can be recapitulated on the chip, right? But it's also incredibly difficult. As I mentioned, for example, we know the blood-brain barrier resides in the endothelial cells. Unfortunately, it's not entirely intrinsic. So for example, if you take a piece of dissociated endothelial cells outside, even the molecule I talk about, MSC2A, disappear within a couple of hours. So there's plenty of results demonstrating this is not intrinsic property, blood-brain barrier. It absolutely needs the local environment to maintain it. How many cell types we don't know, maybe not only limited to 1 or 2 types, right? And also our lab begin to discover there's a heterogeneity even in different regions of the brain. So the local environment, like if you mix the astrocytes or parasite from one area to the other, they may give you different permeability. So I think that's the real challenge for having the chip. I think it's a great effort. It's still worth to try it, but it's very important to constantly compare in vivo situation to see if it's really mimicked or not. It's a great question.
That ground truthing is always important to see.
Yes, ground truth. Yes.
Yeah.
Great.
Wonderful. Well, thank you so much. Wonderful, exciting talk to close on this morning. We're going to take a brief break and restart again at 10:45 Pacific time. So for everyone else, do the math and figure out what time that is you're in your region. During the break, we're going to have a survey. We're going to post some information about future events. And we see you back for the rest of the sessions in about 5 or 6 minutes. Thanks again to all of our morning speakers, and please stay tuned for our afternoon session as well. Thank you.
Great, thank you, Kathy. For those of you who are still in front of your screen during the break, we just wanted to let you know about a couple of upcoming virtual Frontiers Group events that we'll be hosting. The first is Hindsight 2020, the Allen Institute Developmental Recording Symposium. This will be taking place as a series of symposia with the kickoff event happening on November 18th. And then Expanding Minds, the Allen Institute Conference on Human Brain Evolution, will be taking place in January 2021. Information about both of these events can be found at allenfrontiersgroup.org/events. And while we have a few more minutes during our break, we are going to launch a poll that will pop up in your Zoom window. Just want to learn a little bit more about our audience and where you're calling in from and what motivated you to register for this event. So you should see that shortly, and we appreciate you taking the time to answer. We'd also like to send a reminder that there are still questions in the Q&A panel. We will work to have our presenters answer any remaining questions that they may have that are directed to them. So be sure to keep checking the question and answer panel in the answered column. For those of you who are joining us back from the break, we're going to get started in just a couple of moments. We ask that you complete the poll that is on your screen. Just looking for more information about our audience today, where you're calling in from, how did you hear about the event, and the areas of most interest to you. Also, be sure to check out future Frontiers Group events at allenfrontiersgroup.org/events. And we are going to end the poll in just a moment. Thank you to those of you who completed the poll. And this is just another reminder for our next presentations. Please be sure to submit your questions in the Q&A panel. That is where we will be looking for questions during the question and answer portions after the presentations. Okay, and I do have 10:45 now, so I'm going to pass it back over to Dr. Kathy Richmond.
Excellent. Thank you so much, Megan. It's wonderful to welcome everyone back from that incredibly short break. But I think you'll be very excited about our next speaker. I know I am. It's my pleasure to introduce Balaji Kak. Dr. Kak is a professor of physiology and neurobiology at UCLA and is an Allen Distinguished Investigator in our 2018 call. He also continues his transformative work of unmasking astrocyte biology. And he'll be covering how much of those new and recent insights that he's gained, as well as a look to the future. And his talk is entitled Context-Specific and Exploitable Astrocytes. Thank you, Bal.
First of all, good morning to everyone from a sunny but slightly smoky Los Angeles. And thank you to Kathy and the other Frontiers team members for putting together this symposium. It's nice to see many friendly faces from the other speakers on the screen too, and I hope everybody is doing well. What I want to do in the next 15 minutes or so is to split the talk into two halves. In the first half, I want to tell you about a recent and unpublished story where we've explored astrocytes in a specific brain area and discovered that they're remarkably malleable in the responses that they mount, and they have context-specific responses dependent on how they're perturbed, and that it's possible to understand these context-specific responses in molecular terms and use that knowledge to design strategies to be able to exploit these cells for phenotypic benefit in a mouse model of a complex brain disorder. And in the second part, I want to rise to the challenge that was given to us to talk about new areas and present to you an area which I think is ready for growth and could see a significant growth in the next 10 to 15 years, and propose to you how this could be achieved in 4 relatively easy steps. So by way of introduction, astrocytes are fascinating and complex cells. Here's an image of one astrocyte, and you can see it has a cell body, it has several branches, and it's very bushy territory. And these cells exist throughout the brain. They've been known since the mid-1800s or so. Now, the last 10 years has seen a resurgence in interest in understanding these cells, and there's been important breakthroughs throughout the field from many people around the world. But what I'm going to do in this slide is to tell you very briefly about what we've been up to and how this led to the current story that I'm going to tell you about today. So the first thing we started to look at is the structure and function of these cells, how these structured cells look like, and how they contact synapses. And what we found was that these fine processes at the edges of these cells extend out and contact synapses within the striatum with these finger-like projections. And a given astrocyte in the striatum, this brain area that we're interested in, can contact 10 medium spiny neurons and around 50,000 synapses. So the potential for interactions with neurons was very high. The second thing I want to tell you about is the identity of the cells. By comparing these cells between brain regions, we discovered that the dismissive view from many years ago that astrocytes were a type of ubiquitous glue was in fact wrong, and that astrocytes had very strong circuit-specific functions and were actually even diverse within a given brain area. The third thing I want to tell you about is we developed several methods to start to interrogate these cells to determine how they regulate circuits. And we found a variety of mechanisms in vivo to suggest that astrocytes can regulate circuits and drive behavior. And most of these mechanisms happen to be slow mechanisms operating on a timescale far slower than neurons. And the final thing that we did was to explore their contributions to Huntington's disease, because Huntington's disease affects the striatum. And we found that their contributions really reflect a loss of essential functions rather than the acquisition of new deleterious functions. So that's great, but what does this all mean? This really led to 3 further and important questions. The first of those is, how do astrocytes respond at a molecular level to different perturbations? Are their responses uniform, or are they specific to the perturbation? Secondly, can these responses be understood and interpreted, and can you use those to try and manipulate physiology in vivo? And so that's the question those are the questions we want to address. And in order to address those, we had to come up with an experimental strategy. An experimental strategy has three components. We focused our efforts on the adult striatum using tools that we've developed over the last few years. This is important because if you simply analyze data that other people have published previously, you miss out on some of the nuances of astrocytes between brain areas. Then we took those astrocytes in vivo and we challenged them with 14 experimental perturbations that fall into 4 categories: Huntington's disease using 2 mouse models at 2 ages, pathology, for example, ablating the neurons, causing neuroinflammation, a mouse model of OCD, altering ionic signaling, for example, the potassium channel, calcium signaling, and activating distinct GPCRs through these DREADs, GQ, GI, and GS. And then we performed RNA sequencing for all of these and their controls and subjected them to a platform of data mining and analysis. And through the end of this, we generated a hypothesis to see if we could come up with a plausible idea to affect outcomes in Huntington's disease, which we analyzed in multiple scenarios. Now, there isn't enough time to tell you about all of these experiments. So what I'm going to give you is a taste of the key findings. So first of all, what this graph shows are the number of altered genes, red being upregulated, blue being downregulated, and the 14 experimental perturbations. And simply looking at this graph, you can tell that all of these bars look different. And what that immediately tells you is that these perturbations produce very specific effects. Lots of things change when you ablate neurons. Few things change when you have a mouse model of OCD. What this lower panel shows is a heat map where the top 20 genes for all of these perturbations are shown. And again, just by looking at the colors, you can easily discern that there are differences between these perturbations. And we subjected these sorts of data to multiple types of analyses, looking at the number of different differentially expressed genes, the magnitude of the changes, the identities of the genes, their overlap, the gene networks, the signaling pathways, the upstream regulators, and upset analyses for all possible combinations. And at the end of this, we concluded that astrocytes are remarkably malleable and respond strongly in a context-specific manner, depending on how they're perturbed. And I think that's an important conceptual point to make. So this raised the question, do meaningful relationships exist between these perturbations? And to address this, we then moved to analyzing what happens in the context of Huntington's disease. And so this shows you a summary of the signaling pathways that are altered in human postmortem tissue grade 0, which is fairly normal, and grade 3, which is very advanced. And the major pathways that we found in the human tissue is synaptogenesis, protein kinase A signaling, and GPCR signaling. And then we compared these data to what we saw in the mouse models for a whole tissue of the striatum or astrocyte specific. And what we found was that many of the same pathways were conserved. And so this is important because what this tells you is that what we see in the mouse does in fact reflect something that we see in the human. And then we asked, what are these changes in the mouse and how do they relate to the broad 14 manipulations that I described earlier? And what we found was there was one manipulation, activation of a GI GPCR pathway, which produced a reciprocal effect. That is to say, the pathways that were disrupted in the mouse models of HD seem to be corrected by the activation of this pathway in vivo. So to summarize this large bit of analysis, what the second bit tells us is that you can analyze data from humans and mice and come up with pathways that are downregulated in astrocytes and identify a differential pathway which, when you activate, produces a largely reciprocal effect to correct that molecular dysfunction. And so now the question became, can you take this knowledge and exploit it? So to test this, we designed this experiment where we took these mouse models of HD, injected them with AAVs to express this GI GPCR, and then gave them the agonist for this GI GPCR, and then assessed multiple readouts from the mice to see if anything was improved. And this included calcium imaging, electrophysiology, spine analysis, animal behaviors, RNA-seq, and RNA-scope. Again, I don't have time to describe all of these, so I'm just going to give you a taste of some of the key observations. So it's well established that in Huntington's disease, EPSCs mediated by glutamate are reduced. And so the first thing that we found and we assessed was these. So these are inward AMPA receptor EPSCs. These are outward NMDA EPSCs. And in the mouse model of this disease, these are reduced in amplitude. If you activate the astrocyte GI pathway, we found that these were recovered. This recovery was complete for the fast synaptic excitation, and there was a slight overshoot for the NMDA component, and there was no change in the paired pulse ratios. So we've assessed other things too. For example, we see a recovery of the mini EPSCs through this astrocytic intervention, the recovery of spine density, a recovery of astrocyte calcium dynamics, and a recovery of astrocyte morphology. I would, of course, be remiss if I didn't tell you about things that didn't recover. And so there are things that don't recover. For example, that we didn't find a recovery of MSN excitability, the properties of the action potentials, tissue loss, and dendritic morphology. And I think actually knowing things that do recover and things that don't recover is a feature and not a bug. Because what this tells us is about the things which are most proximal to an astrocytic mechanism and those which are non-cell autonomous and driven by neuronal changes themselves. Nonetheless, buoyed by this data, the next thing we did was to see what happened to behavior. And so in Huntington's disease, there is a well-defined set of behaviors that can be assessed in vivo. And these are summarized in this heat plot. And the basic take-home message is that when you activate this GI pathway in astrocytes, you can correct many of these behaviors and produce phenotypic benefit. But I want to go into a couple of the details because, again, the details are very important. So in this one behavior of the open field test, which essentially measures the ability of the mouse to move around in an open arena, you see a deficit with HD in the models, and this is completely recovered by activating this astrocytic pathway. This is true also for self-grooming. This is the ability of the mouse to engage in this OCD-like phenotype, and this is completely recovered. But there are other behaviors such as rearing, which, when a mouse rears and looks around, it's sort of its alertness, its awareness of its environment. This is partially recovered and not completely recovered by an astrocytic mechanism. And there are other phenotypes like hind limb clasping, a classical phenotype of these HD mice, which are not significantly recovered at all. And so what this tells us, I think, again, are those mechanisms which are proximal to astrocytic mechanisms and other mechanisms which are driven by additional changes within the neurons themselves. So, to summarize what I've told you so far, astrocytes are remarkably context-specific and change in separable ways depending on how they're perturbed. These changes can be understood using bioinformatics, and we identified a reciprocal change within a parallel pathway. We exploited that pathway in vivo, and what we found was that you could correct several phenotypes associated with HD at the level of the astrocyte, at the level of synaptic function, and at the level of mouse behavior. And what we've done subsequently is to perform an additional set of molecular analyses following the activation of this pathway. And what we find is that the major mechanisms that drive this are related to the neuroimmune system, neuroinflammation, and synaptogenesis. So this is why this happens through an astrocytic mechanism. And by mining these data, we've also found 23 native GPCRs, which are enriched in astrocytes found within the striatum. And I think these 23 GPCRs are very interesting candidates to explore further biology of these cells and also potentially, who knows, as potential therapeutic targets. So now I wanted to switch and tell you about an area which I think is ready for growth. And I've called this area integrative brain cell biology. And to illustrate what I mean by this, I've shown you this cartoon. And what this shows are all the different cell types in the brain. So of course, neurons are very important. They represent about half the cells of the brain. But the other cells of the brain comprise two major classes: glia, which consist of astrocytes, microglia, and oligodendrocytes, and the cells at the brain borders, which we've heard about this morning, including immune cells and the cells that form the blood-brain barrier at the meninges and at the neurovascular unit. It's fair to say we know less about these cells than we ought to. And so I think an interesting area for the future is to unmask the biology of this, the other half of the brain. And I think this is important to achieve as a discipline, because it would allow us to begin to understand the brain as a highly evolved multicellular organ rather than simply as a collection of cells that we study one favorite cell at a time. And so how could one go about doing this? And I think there are 4 ways you could do it. So what this image here shows are astrocytes in the hippocampus, where there's 10% of them labeled, 40% of them labeled, or 90% of them labeled. And the only point in showing you this is to illustrate that these cells at this type of imaging essentially look the same. But a big realization in the field has been is that glia are actually remarkably diverse between brain areas and diverse within a brain area. And so this idea needs to be chased down and quantified throughout the nervous system so we can get a handle on this molecular identity and molecular diversity. Of course, doing this just by sequencing isn't enough. We need to understand what that means functionally for specific brain areas and within brain areas. And of course, we need to do this in mouse models and in human tissue. If we could do this, then I think we could place the field on a quantitative footing to be able to perform hypothesis-driven experiments. The second thing I think we need to do is illustrated here. So what this shows you is a microglial cell, an astrocyte, and an oligodendrocyte. And you can see that these cells are all remarkably complex. And if you take a step back from this, I think there's an important point here. And that is, whereas neurons seem to have evolved to perform their core functions at distance through extensive axonal and dendritic arborization, non-neuronal cells seem to evolve to perform their core functions locally through highly complex structures. And I think the trick to understanding what these cells do is to understand what happens in these small structures on the submicron scale. And so what we need to do is we need to understand signaling in these small domains and understand how these cells talk to each other, what proteins are expressed in these small domains, what the mechanisms are of these small processes moving relative to each other, and understand the structures of these cells at high resolution, both for mouse models and in humans, and determine how signaling is organized. If we could do that, then we would have a cell biology of the non-neuronal cells of the brain. The third thing I think we need to do is summarized in this slide. In the last few years, there's been tremendous progress in understanding how glia regulate neurons. For example, Beth Stevens' lab has shown microglia contribute to cognitive decline in AD. Mark Freeman's lab showed that astrocytes contribute to multisensory behaviors in fruit flies. And Nelson Spruston has shown astrocytes contribute to barrage firing of interneurons in the hippocampus. We've shown astrocytes contribute to OCD-like repetitive behavior and ADHD-like behavior. And Inbal Goshen's lab has shown that astrocytes contribute to fear memories. And Misha Aaron's lab has shown that astrocytes contribute to passivity and giving up in zebrafish. If you put all of this together, what this suggests is that glia may contribute to cognitive function and may explain aspects of brain function that can't be explained by neurons alone. And so there's a real opportunity here to understand how glia regulate synapses, how they regulate neurons, and how they regulate circuits. I think the key question here is to ask, what is it that glia impart to the function of a circuit? When is that function used, and what is it good for? And addressing this question will require glial biologists, but also neuroscientists working together hand in hand to see how this biological layer of complexity works. And it will require systems biologists and computational scientists. Now, the fourth topic is illustrated here. This is a drawing taken from 1911, and these cells we would now recognize as glial cells surrounding plaques. This is an image from my lab, where you can see a plaque in the center surrounded by astrocytes here. This is an image from my good friend Beth Stevens' lab, where you can see a plaque surrounded by microglia. We've known about these sorts of things for many years. So there's an association of astrocytic and glial dysfunction in a variety of diseases. And these sorts of cellular data are now buttressed by single cell sequencing data where we know these cells are disrupted. But there are many questions. What are the correlative and causative functions of these cells? Like, do glia simply change, or do they actively contribute to the disease process? And I think this question, it needs to be addressed, and if it could be addressed, may be meaningful. In order to address it, we need to understand how these signaling mechanisms change and drive disease phenotypes. And we also need to develop what you might call an end-to-end biology of these cells, how to go from molecules to a translational platform in vivo. And I think how to do that for a population of cells about which we know so little is itself a challenge that needs to be embraced at some level by the field. And finally, can we exploit this knowledge to develop glial-based therapeutics? So as you can tell, the study of non-neuronal cells is in a very exciting field. It's certainly a field with many open questions that need to be addressed. But the one question that some of you may be wondering is why now? Why not in the past? Or why not at some undisclosed time in the future? Well, this question is remarkably easy to address. We're dealing with a large, but still largely understood cell population. There's been a distinct lack of tools to study these cells. Those tools are emerging. We'll need to generate more, but those tools are beginning to tell us that these cells are important. There's new data on cell diversity rejecting the notion that they're simply a glue. The structure of these cells is completely unexplored. There's evidence for roles in complex brain phenomena and also in disease. And if you put all of this together, then I think what we have before us is an opportunity for new discovery, for training a new, diverse, multidisciplinary population of scientists, and potentially for therapeutic approaches. And I think this could be done in 10 years by 5 labs working in a largely unfettered and unrestricted manner. And I'd finish by thanking the Paul G. Allen Family Foundation, my other funding, my collaborators who did the bioinformatics, Madan Babu, Miriam Arti Solano, and Giovanna Coppola, and my lab. This is in the early March, especially 2 people, Jun Nagai and Jinzi Yu, who have now left to start their own labs. I'll stop there and I'll be delighted to take some questions.
Excellent. Thank you so much, Paul. Wonderful, wonderful view of a topic that is just now, I think, really generating so much excitement. And some of the questions, I think, reflect that. So one of the questions that we have is, how dynamic are astrocyte populations, and are they long-lived?
So dynamics could mean different things. So if you look at their signaling, they're very dynamic in terms of their intracellular biochemical signaling. The best version of this is calcium signaling. They have thousands of little sparks. When we found these using the latest methods developed by Roland Ruger 10 years ago, I call this a sparkling panorama of calcium signals. It very much looks like that. And so they're very dynamic at the seconds time scale. Also at the minutes time scale, and possibly even at the seconds time scale, they can extend fine processes towards synapses, do something, we don't know exactly what they do, and then move away again. So they have spatial dynamics. Of course, they're very dynamic in the context of injury. If you were to cause a lesion or an injury, or possibly even a molecular injury, then glial cells can move and they can surround that injury. They can sort of surround it and form, in some instances, what's called a scar. So yes, they're not a static population. They're dynamic at multiple scales and at multiple biological levels of interrogation.
So are they long-lived?
So I think they are long-lived. I would be speaking out of turn if I gave you an accurate number, but I think some of these studies have been done following the nuclear sort of accidents that have happened around the world and looking at incorporation of heavy isotopes. And from those types of experiments, you can tell that they are long-lived, yes.
And then another question asks, to what extent do they differ between species? Are there other model organisms that could have advantages for use?
Yeah, absolutely, there are. And we've, several of us during the COVID shutdown, 10 of us in the field have got together and done a sort of meta-analysis of glial cells in human, in mouse, in zebrafish, in Drosophila, and in C. elegans. And it's remarkable that some of the mechanisms that exist in rodent are actually shared across these organisms. And so I think this really is an evolutionarily ancient mechanism where the core biology is conserved across species. And there are tremendous insights from C. elegans, from Shai Shaham's lab, from zebrafish, from Kelly Monk and Misha Ahrens and Mark Freeman, many others in Fly that are strengthening this idea. So yes, this should be done in other species. It's just that the time wasn't sufficient for me to explain all of that work.
I'll just expand a little bit on from that, because you noted a lot of model systems I think people may be familiar with. What are your thoughts about outlier systems, whether it be ones like Hydra or Octopus or other ones that people are using to maybe come in it with more frontier approaches or novel approaches to ask questions about the nervous system or that ecosystem.
Yeah, that's a great idea. I mean, those are species that I don't know much about, but I think it'd be worthwhile taking a look. Even if there weren't bona fide astrocytes as we define them with the metrics we know, it's quite possible that they have another cell type that may look slightly different, that subserves a similar function. So I think there would be great value in looking at additional species and doing this sort of analyses across different organisms.
So here's another question from Martin Frasch. Fascinating notion to get the submicron level of glial cells complexity. What technologies do you anticipate or know exist already to help us do that?
Oh, so there are methods that we could make a pretty good start on this. I mean, I didn't have time, but there's some great work going on at the Allen Institute from Clay Reid using special types of electron microscopy. And there's great work going on at Janelia using focused ion beam scanning electron microscopy. And the latest methods on these can give you a sort of isotropic resolution on the order of 4 nanometers or so. I mean, they're intense. There's a lot of data analysis to do, so one shouldn't underestimate the task at hand. But the methods are giving one the resolution that's needed to see what's in the fingertips of those fine processes and how they're organized and how their organelles are organized inside them. So I think that's definitely feasible. As far as I know, there isn't any work on this at all on glial cells. There's certainly nothing published and at the resolution that's required, but it's doable with the right type of effort.
Wonderful. Thank you so much, Paul. I think you'll have a slew of questions to answer on the Q&A.
Okay, great. I look forward to it.
Really,
Thank you very much.
Thank you so much. And our sixth and final speaker for the session is Han-Kui Zheng, our Executive Vice President and Director of the Allen Institute for Brain Science, and that'll be our last talk before we start our fireside chat. Dr. Zheng has been at the Allen Institute since 2006 and has led the development and operation of high throughput pipelines to generate large scale and open access data sets and tools to accelerate neuroscience discovery. It's my pleasure today to introduce her, and her talk is titled Understanding Brain Cell Type Diversity. Han-Kui.
Thanks, Cassie. Can you hear me well?
Yes.
Hello, everybody. First of all, I want to thank Kathy and the Allen Frontiers Group for organizing this wonderful event and for inviting me to participate. In this talk, I will give you a broad stroke overview of our effort at the Allen Institute for Brain Science in trying to gain a systematic understanding of brain cell type diversity. And in response to Kathy's request, I will also give you some personal perspectives in terms of what I think the neuroscience field should and can go in the next 5 to 25 years, taking advantage of the vast amount of knowledge that we have been gaining in cell type diversity in the brain, and how the Allen Institute can contribute to that endeavor over the next several decades. So to start with, I think to understand how brain works is truly a noble goal for at least 2 major reasons. Number one, it is to satisfy the human's curiosity in trying to understand brain, this arguably the most complex system in the known universe, how it generates behavior. And not only that, but the human intelligence, emotions, and creativity. And having this kind of knowledge and understanding can also in turn inspire our own creativity in developing new inventions to improve our life with the neural network-based deep learning approach as a great example of that. The second major reason is that by understanding how brain works, we then will be able to develop better ways to treat and alleviate the devastating effect of the many brain-related illnesses. However, how do we do that? Given that brain is such a tremendously complex system, we think that we need to take a multi-level approach to decompose this kind of complexity. For example, this slide showed a four-level approaches in gaining hoping to gain a mechanistic understanding. We want to understand the components of the brain, namely the diversity of the cell types in the system, both neuronal cells and non-neuronal cells. And then we want to know how the cells interact with each other. For neurons, this would be the wiring diagrams among the different cells and cell types across the entire brain. And then we can see how the different types of cells function in animals, in humans and other animals, how they behave under different behavior conditions. And finally, we can develop computational models and theories to try to explain and possibly predict how the networks and circuits in the brain can generate behavioral and other outcomes. So at the Allen Institute, one of the major goals is to work on even just this first level, trying to understand the composition of the brains, the cell type compositions, and the diversity of the cell types. And this is because, over the last 100 years, ever since Ramon Cajal, we have known that in the brain, both the neurons and now non-neuronal cells have tremendous heterogeneity and diversity in the different types of properties they have, from molecular properties to morphological, connectional properties to their physiological and functional properties. And it's those diverse properties of the individual cells and population of cells that allow them to flexibly work together to exert a wide range of behavior. So at the Allen Institute, we have really taken a battery of large-scale standardized approaches to profile cells at the individual level and use those information to define cell types, systematically categorize them, and use cell type-based knowledge to investigate the connectivity and function of the cells in their embedded circuits. Here, I'll just briefly overview the kind of approaches that we have established at the Institute. First, we take this highly scalable single-cell RNA sequencing approach to profile thousands, tens of thousands to millions of individual cells in different parts of the brain, and use a data-driven approach to generate, to cluster the data and generate taxonomies of cells and cell types. At the other end of the spectrum of the approach that we're taking, we investigate the connections of neurons across different parts of the brain at both brain-wide mesoscale level and local microscale level, using different anatomical and physiological approaches. And we do that for both populations of cells, in many cases using genetic tools to target specific types of cells. And we also investigate that the connectivity at a single cell level, both brain-wide and at local circuits. And we look at the connections between cells at both anatomical levels and as well as at the physiological levels. In addition to that, we also, like many other labs in the world, look at the activities of different cells and types in awake behaving animals using in vivo activity imaging or electrical recording approaches. To bridge this vastly different types of information at the molecular level and at the system level, we take several different approaches to do that. We generate cell type targeting tools, both transgenic animals, as well as enhancer-based viral vectors, to allow us to label and manipulate specific cell types. We also take a multi-model integrated approach using a technique called patch-seq to obtain molecular, physiological, and morphological information from the same cells and profile thousands of cells in this way to be able to establish the relationship of the different types of properties of the cells and use that information as a bridge to link molecular identities of the cells with their physiological, connectional, and functional properties. We also use spatial transcriptomic approaches such as a multiplex FISH in situ fluorescent hybridization approach to not only look at the spatial distribution of individual cells and types, but also use that as a post hoc method following the connectional and functional studies of the cells to decode the molecular identities of the cells that we have obtained connectional and functional properties from. So with this comprehensive set of approach, we have now been generating really a large set of data, starting from specific cortical regions in both mouse and human brains, to really understand the multi-model diversity of the cell types. And we are now expanding that work into multiple different regions of the brains in both mouse and humans. And this work in particular was supported by the Brain Initiative effort. We are now part of this large community that's supported by NIH called Brain Initiative Cell Census Network, where together with dozens of other labs around the country and around the world to use the same kind of approaches that I described in the previous slide to systematically characterize individual cells using a variety of different approaches and in multiple mammalian species in order to build and arrive at a cell type atlas, multi-model cell type atlas across the entire brain and possibly in the future across multiple species. So the knowledge we have been gaining from this tremendous effort, collaborative effort, would include molecular taxonomies of cells, brain cells, across multiple mammalian species, the spatial distribution of those different molecular types using spatial transcriptomic approaches, the multi-model integration of the different cell types, again, using patch-seq-like approaches, and the connectivity or wiring diagrams of the different types of cells within a particular region and between that region and their input and output brain regions. So in the next couple of slides, I will just give you a little bit of highlight of the major data types and conclusions we have been able to obtain from this kind of a large-scale effort. The first front-end approach, as I said, is large-scale single-cell transcriptomic profiling that is able to give us an initial framework of cell type classification in a particular region, and hopefully in the very near future, for the entire brain or the entire nervous system. So this highly scalable approach allows us to profile, as I said, up to millions of neurons pretty rapidly. And we classify the cells into types and to generate a taxonomy of cell types. We primarily use two major approaches, the deep sequencing-based SMART-seq approach and the 3' end sequencing droplet-based highly scalable multiplex approach. And in our particular case, we used a 10x genomics platform. In our published work, we have used the SMART-seq deep sequencing approach to profile two separate cortical regions in the mouse and one cortical region in a human brain. And we found a high degree of conservation of major cell types across those two major mammalian species. At the same time, we also find species-specific, a high degree of species-specific gene expression profiles within each major types, illustrating both conservation and divergence, important features of cell type comparison across species, which would have important functional implications. And in the interest of time, I will not show you more results from these amazing technologies. Just wanted to say that we have now been able to expand our work into profiling millions of cells, together with other scientists in the BICCN consortium, to profile now the entire cortex of the mouse, as well as many, many parts of the subcortical regions. And we now expect that we will be able to obtain a transcriptomic classification for the entire mouse brain, the entire mouse central nervous system, over the next couple of years, just the next 1 to 2 years. So it's a very exciting time to look at the data and understand the relationships of the different cell types now. Another major study we have done is to compare and combine the transcriptomic profiling in the individual cells and epigenomic profiling of the cells. And from there, we hope to be able to gain further knowledge of the cell type specific gene expression landscapes and mechanisms. In collaboration with several groups within BICCN, we are able to combine 8 different data sets for one particular mouse brain region, in this particular case, the mouse primary motor cortex, using integrative computational approaches to cluster this vast set of disparate data together to come up with an integrated transcriptomic and epigenomic classification of the mouse motor cortex cell types, in particular neuronal types. And we find that this integrated classification is highly consistent with each of those 8 individual RNA sequencing, or methylation sequencing, or ATAC-seq data types. And because of this high degree of concordance and the integration of the different data together, we are able to reveal, indeed, the epigenetic landscapes in a cell type specific manner, in a truly data-driven cell cluster specific manner, and correlating specific gene expression with the availability of the chromatin elements within the same cell type. And from there, identifying thousands of concordant regulatory elements, potential regulatory elements that can be used to not only study the mechanisms of the cell type specific gene expression regulation, but also used for the generation of cell type specific targeting tools. In a third type of study that I want to show you about is a multi-model integration of the different properties of the individual cells through the patch-seq approach. In this recently completed study, we profiled more than 4,000 individual cortical interneurons. This time is specifically from the mouse visual cortex. And again, using a data-driven classification approach, we are able to integrate the transcriptomic types and the morphoelectrical types together to form a whole new set of what we call MET integrative cell types, in which the cells belonging to each type exhibit highly concordant transcriptomic, morphological, and electrical properties that are also mutually predictable, with high degree of mutual predictability. So here shows the 28 different MET types that we have identified for cortical interneurons that cover all the major known and novel cortical types. This kind of integrative information is extremely important in bridging the gap between the molecular identities of the cells and the morphophysiological functions, properties and functions of the cells when they are obtained in separate studies independent from each other. And in the final example study I want to tell you about is this large-scale, really heroic, EM connectomics approach that we have been taking at the Allen Institute led by Clay Reed, as Bojit mentioned earlier. We have established EM pipeline at the Institute using 6 different TEM, transmission EM systems. We developed a highly scalable sectioning and imaging approach that allows us to image very large volumes of EM data. Large volume in the world of EM is, let's say, 1 or a couple of cubic millimeters, which comprises of at least 25,000 sections, each 40 nanometer thick. And with nanometer, synaptic level resolution. So here shows an example, 1 cubic millimeter volume that we have collected from the mouse visual cortex using this large scale approach. And through advanced machine learning approaches, this large volumes, petabytes of image data can be automatically segmented and individual cells from the volume reconstructed so that we can identify them as different morphological types and also gain their synaptic connectivity information. Furthermore, through the study that I just showed in the previous slide, the patch-seq MET type of study, we can then also infer the molecular identities of those cells in the EM volume through the correspondence between molecular profiles and the morphological features of those cells. So with all these different approaches integrated together, as you can see, with transcriptomic, epigenomic, multi-model, and high-resolution connectomic approaches, we're now truly at a stage where we can gain comprehensive understanding of the diversity of the cells in brain. So where do we go from here? I just gave you a summary of what we can achieve today. How to harness this tremendous knowledge that we are gaining to help us go to the next step, to understand the brain function. In the next 4 slides, I will just give you some of my perspective of what we can and should do. The immediate thing we can do, I propose, is that we can take a systematic approach to bridge the knowledge about cell types and brain function. And one of the main things we can do is to construct what I call a molecular and anatomical architecture for the mammalian brain. And when I say a molecular and anatomical architecture, it means a combination of molecular cell types as identified by omics approaches with the spatial location and the shape and morphology of the cells, and with the connectivity of those cells in a brain-wide manner. So that entails, for example, the generation of a brain-wide cell type map, which would contain molecular identities of all the types and possibly even in single cells, the spatial distribution of all those cells and types across different parts of the brain, so to understand the spatial organization, the connectivity of those cells and the types at both population level and individual level, and to obtain targeting tools, as I showed in a slide earlier, that allows us to target, label, and possibly even manipulate those different cells in different parts of the brain. And finally, generating this kind of a brain-wide cell type map, not just for mouse, but for multiple species, and looking for similarity and differences across species so that we can understand both generalizable universal principles for the brain organization, as well as specific species-specific specializations. I think this, having this kind of architecture will be tremendous powerful for us to begin to really understand how brain works by having the different parts of the brain working together. And it's not only powerful, but also more excitingly, it's doable. It's really, really doable now. So I'm very excited by that. The second aspect we can look into using the cell type knowledge that we have gained is to begin to understand the development and evolution. We know that cell types arise from elaborate evolutionary processes across different species, as well as from highly complex developmental processes. So there are evolutionary lineages and developmental lineages that lead to the current complexity of the brain cell types. By studying the developmental and evolutionary processes of the cell types, we can not only understand the cell types themselves better, but also understand how the brain is built through this developmental and evolutionary processes. We will be able to know better the comparisons across different species, as well as the entire developmental processes and trajectories, establishing this kind of complete lineage trees using advanced lineage tracing approaches coupled with single cell omics approaches, and understand not only where each cell type comes from, what mechanisms control their fate at an eventual identity, but also how the cells interact, how the system interacts with the environment, how the environment shapes this process, and what are the relationships of the different cells across different regions and different systems. Finally, all of the studies will really tremendously benefit our endeavor in eventually understanding and treating a variety of brain diseases, throughout the lifespan of an animal or a person. The different types of illnesses, they manifest themselves in very different ways. And by having a foundational knowledge of cell types, it will allow us to understand better exactly what's happening in the devastating diseased conditions. From the many previous talks you've heard, that we can begin to understand better the genetic, epigenetic, somatic, genetic underpinnings of the different kinds of diseases, and the potential selective vulnerability of the different cell types under different disease conditions. Cross-species comparisons of the different cell types will allow us to also evaluate the existing disease models and build better and more predictive disease models. And finally, to come up with cell type and circuit-based therapies, many examples as shown by the previous speakers. And here's my final slide. I think that, at this age, Kathy was asking me to think really boldly about what we should do. And I think it's true that it's good time with the technology advancement, it is a golden time now for us to have bold moonshot kind of thinking, and to work together on breakthrough projects. I'm talking to you, John Nye here, as the new director of Brain Initiative. It doesn't have to be a single, monolithic moonshot projects, but we have to have the kind of moonshot type of thinking. Think big and bold, and think about how we can work together. And I think this is really the current thinking of many of the previous speakers as well. So just going back to the complexity of a brain, the multi-levels of complexity, and how can we really work out, to figure out how brain works. To crack the complexity of the brain, I'm not proposing specific projects here. I'm just giving you some general thoughts that I have. I think it will, in addition to many individual PI-driven, hypothesis-driven research that's taking place, we should also have large-scale, multidisciplinary, collaborative approaches and projects, like what we have been taking at the Allen Institute, and like what's happening at the Brain Initiative Cell Census Network. And I really realized that this kind of a multidisciplinary, collaborative approach is actually really highly fruitful in generating new insights, unexpected new insights, by looking at the data in an integrative way. And it has to be open science, open sharing of data, and global coordination, public-private coordination would be really critical in putting all our effort together. And then scientifically, just like what we have been doing in the cell type field, I think that complete, accurate coverage is critical for us to identify generalizable principles and to have an unbiased view, and to discover, to explore and discover. There are a lot of beauties in the biological data themselves. And when you see everything together, you will find new things. And finally, of course, given the multiple levels of complexity, as I mentioned earlier, and given that we will be able to generate this kind of a complete, accurate data set, multi-level, multi-dimensional integration will be critical. Advanced computational approaches will be critical to allow us to integrate the different kinds of data together and to obtain insight. Okay, so with that, this is the end of my talk. I want to thank our founder, Paul G. Allen, for truly inspiring effort in inspiring us to do different things, to explore new frontiers, as Cassie mentioned. I also want to thank NIH, multiple institutions of NIH, in giving us grants to support our work as well. And finally, the large collaborative teams at the Allen Institute that makes all this work possible. Thanks for your attention.
Excellent. Thank you so much, Han Kui. A wonderful overview and wonderful look to the future. I know that we're running a little bit late, so your homework is to answer all the questions that have come in after the fireside chat. It'd be wonderful to start the fireside chat right away, and you've already somewhat teed that up. It's my pleasure to introduce both you and Dr. John Nye, the director of the NIH's Brain Initiative, and of course, for those of you who may not know, BRAIN is an acronym in this case that stands for Brain Research Through Advancing Innovative Neurotechnologies. And he oversees the long-term strategy and the day-to-day operations of this groundbreaking enterprise, and will be joining us for a chat. Really look to see again what the future holds. Now, I'm going to pose to both of you. It's been really exciting during this session to see all the different ways in that these thought leaders have come up with and what the future may look like. I was curious if there were any surprises, if you thought that there were any things missing, what you thought of what the future holds and your own views of it.
Shall I start?
That'd be wonderful, John.
And I think it's been a terrific morning of science. I think great science means we should always be surprised by answers that some of which we might have predicted, but never quite in the way that it plays out. And I think that's the beauty of pursuing discovery-based science. It's the curiosity that we have to pursue questions, not just for the sake of getting that answer, because that is in and of itself satisfying as scientists, but also how can we turn that into some benefit for society. I think some of those benefits are longer time in coming than others, but it really what makes the scientific enterprise so much fun. I think we've seen some amazing discoveries discussed here today that some of them were enabled in part by great technologies, but they were driven by people asking the right questions. And I think that's really, I think that's a great take-home message for today.
Yeah, I just want to say I fully agree with that. Yeah.
I know that it's really amazing. Both of you have been enmeshed in this field for decades. And so both of you have seen just how far things have happened in a relatively short period of time. So going forward, I wasn't sure if there were things that you have on your radar that 10, 20 years from now you see as really popping up or bubbling up as to where we may be able to place that stake in the ground.
Quite please.
Yeah, so I just wanted to reiterate what I said, what I proposed in my presentation. I think one of the greatest opportunities, especially built on top of what we have been doing in the cell types, is to generate this what I call the molecular and anatomical architecture of the entire brain that combines molecular information with connectional information. So I strongly support a brain-wide connectomic effort in generating a systematic wiring diagram between cell types and individual cells, all in one shot for all the different regions together. So you can have a overview of the different circuits and the rules in governing that wiring diagram in the entire system. I see that as one of the greatest opportunities we have. As we have already seen, this kind of wiring diagrams that are being generated in several invertebrate species has been transforming our understanding of the circuits.
Yeah, if I can just play off of that, I absolutely agree. The efforts that have been going on for the past several years on developing a complete or a comprehensive cell census of cell types, not just neurons, but also non-neuronal cell types in the brain, together with comprehensive connectivity maps, high-resolution connectivity maps, really will provide the ground truth for not just for further discovery, but for the basis for finding new therapies for curing human brain disorders, including neuropsychiatric disorders. I mean, Hongwei, you really wrapped this up quite nicely in your last slide in looking forward towards circuit therapies. I mean, many of the neuropsychiatric diseases that we face as a society, most of them are complex from a genetic point of view, and we really will have to understand the circuits that are affected, and using these tools of discovery, we will, and using these tools plus other tools that are being developed here in the Brain Initiative as well as outside, for example, in gene editing. Amazing recognition today for that from Stockholm today. We really do have a chance for changing the course of the treatment of human disease in the next, I think within our generation. It's really quite exciting to do.
I mean, given all the different directions that were talked about today and the blending of fields or the multidisciplinary efforts, I'm curious what advice you'd give to students entering the field or people considering entering this field.
The one advice I give to all students and to myself and to my colleagues is follow your passion. If you're not passionate, you're just not going to fulfill your potential. There's so many great questions out there. Choose one that gets you out of bed in the morning and keeps you up at night. And that's probably the one formula for success that I can think of.
Yeah, one advice I would like to give is that the multidisciplinary knowledge is really important. I think nowadays, scientists being able to have a deep expertise in both experimental science and computational science is going to be really powerful for the person to be able to ask and address questions in an informative, efficient way.
Being based in Seattle, it has been exciting to see the role that data science and statistics and machine learning and artificial intelligence have joined the conversation, given the complexity of data that's coming at us with all this new technology. So I know that that'll be an exciting addition to add to the teams that are going forward and to the labs that are going forward. I know that one of the things that we had talked about a little bit was, well, it's hard not to talk about the current pandemic state that we're in and the impact it's had on the research community, but also the inspiration that maybe can be had by looking at the response. So I'm curious what stories you may have or what insights you may have on how that shaped research now or going forward, how we view it and the possibilities. John, we'll start with you.
Yeah, it's been challenging for sure. It's upended life as we know it on multiple fronts. In addition to the personal and societal impact in terms of science, science literally, in some ways, ground to a temporary halt. But you can't keep a bunch of creative scientists on the bench for too long. And very, very quickly, scientists out there pivoted. They took their know-how that they developed on their specific project, and they thought, how could they apply that to find ways to either diagnose or treat and basically knock down the COVID epidemic. And it gets to what Hong Kui mentioned about the importance of multidisciplinary science. If one has access to or has an open mind toward areas of science and technology that are outside one's own training, it opens up a whole new world of possibilities. And I think we saw that. It's really amazing how quickly the scientists around the world pivoted to attack this problem. I mean, we're seeing advances that previously were unimaginable in the timescale that we've seen. And I think that's due in large part because of the creativity of the science, but also because of the technology that enable us to do these projects in a way that we couldn't do it before. But it still boils down to creativity to get these things done.
Yeah, I totally agree with John. I think it's amazing to see the scientific field getting together and in trying to solving this problem so quickly and so comprehensively from all angles, right? From all angles. One point I just wanted to add is that I think it makes the society, people in the society appreciate science a lot more. You know, appreciating that scientific research can bring true difference to their life. This is, you know, a direct testament to that.
You know, another aspect. I know many of the speakers talked today about the blue sky, the new frontiers, the emerging topics that are just ripe for advancing and shedding more light on what health and disease may mean. One of the things that they touched on, though, in addition, was how we get there. The mechanisms, how to cover the valley of death, how to target basic science, whether it be through the type of funding or the pipelines and the like. And I'm curious about your thoughts on that looking forward as well, the role that can play.
I think this is probably a question, a very good question for John as the new director of Brain Initiative. I think funding decisions and also would be really critical, and also NIH can play an instrumental role in organizing the scientific community together, as we have been actually doing within the BICC. So maybe John can talk more about that.
Yeah, it's a great question. I think there are different roles for different styles of science to move the field forward. Ultimately, we look to our own creativity to pose the questions that we set out to answer, and that also is the mother of invention. And what we've also seen over the history of science is that people will ask great questions, they'll use the tools at hand to start getting answers, and then ultimately, one hits a wall. ultimately one finds that in order to probe deeper, to get the answers, not just to those questions, but ways to ask better questions, one needs a breakthrough in how one studies these questions. And that leads to more questions. So I think this is where the Brain Initiative can really help, not in terms of not only supporting great discovery-based research, but really what are the tools that we need to get us beyond these barriers? And some of the tools, I think, is well exemplified by the project that Honghui and others at the Allen have led so successfully, do require team science. There comes a point where you just can't get these things done with individual labs. I mean, sure, individual labs can provide the disruptions, the ideas, but at some point one needs to put these projects into action. So at the Brain Initiative, we're looking at supporting both sides. They're both highly critical. And the key thing also is to get these things done in a timely way. And certainly the tools that we've developed over the last 5 years are allowing us to pursue these projects at a rate and at a depth that we couldn't even imagine 10 years ago. So it's really a very exciting time to do this. I mean, when this country decided to put a man on the moon, the technology had to be developed to get that to happen. And the technology that came out of that goes far beyond that incredible accomplishment in and of itself. I think the projects that Honghui is talking about here, comprehensive cell atlases and connectivity diagrams of complete mammalian brains, frankly, we don't yet have the technology to do that today. We cannot solve tomorrow's problems with today's technology. I think this will be the driver to get it done. I have absolute confidence that we will develop the technologies to get these projects done, and then we'll see how we can apply that directly to human health.
Well, I can't agree more. It's been exciting to see the public-private partnerships and all the ecosystem of funding and the leading role NIH has taken to move a lot of these advances forward. And there is a question that's coming from the audience on how new technologies to look at human brains will deal with the heterogeneity in humans. What's on the cusp, or what's on the cusp, or what should be something that we should be trying to target. And I'm curious, Honghui, you're shaking your head. Are there things that are top of mind for you that can address that?
This is a very important question, the human diversity, individual diversity. I think we just, I don't have a solution for that. It's a tremendous challenge, and we just have to do it. What we are doing now is just really, setting up the foundation, giving you one version of, for example, the cell type catalog. But as Rusty Gage was talked in his talk, there's not only individual diversity, there's even cell-to-cell diversity. Maybe John has more insights into it. This is a major important question, and I don't know how else to address it other than collecting data and compare them, and to see exactly what the diversity is and where they come from.
Right. I mean, diversity is the fruits of evolution in Mother Nature, and it's what makes us different as individuals, but also common as humans when we think about the human condition. As biologists, most of us were brought up to want to eschew or avoid issues of variation. But in fact, I've come to take a different approach. I think variation teaches us about the biology. And it's a huge challenge for us to confront, because it's hard enough for us to understand how the brain works in an inbred mouse. How are we possibly going to do this in a way that really accounts for the incredible diversity across the human species? But I think great scientists respond to great challenges. And it's a good reminder that the Human Genome Project was launched, say, 20 years ago. It cost over $3 billion to get the first draft genome. We can walk into our lab and sequence a human for under $1,000 today. So I think this just tells us that with the acceleration of technology, we shouldn't be afraid of such challenges. We should embrace them and figure out ways to solve it. I mean, the only way to solve these problems is to take them head on. So, well, I don't think we have the answer for how we're going to do it, either in terms of an operational strategy, but also in terms of what technologies we're going to have to develop to get there. But I know that we have to lay that challenge out. We cannot back away from it, because if we do, we'll just fall short, and that will be disappointing.
I know this will be a potentially challenging question to ask, because there are so many possibilities. But what gets you most excited about the future of human brain research? Is it the potential? Is it the technologies? Is it the endpoint? It goes back, I think, to your passion point, John. You have to be passionate and excited about what you do. So when looking ahead and looking downstream 10, 20 years from now, what is the most provocative or exciting target that you're thinking about?
I think for me, it boils down to two overlapping areas. I mean, one is, as Honghui mentioned, the human brain is the most complex system that we really know of. If we can understand how this works, I mean, it would just be a tremendous accomplishment. Or if we can understand just even a little bit more how it works, it'll be a huge accomplishment. The other part is, I think we really do have the potential for curing or treating some devastating human neurologic and neuropsychiatric disorders within our lifetimes. This to me is again, just tantalizing. And I'm always reminding myself that certain things we're doing today, when we talked about them, if we even knew to talk about some of the issues or the discoveries we're making today, 10, even 5 years ago, it was science fiction. And I remind myself today, science fiction is tomorrow's future. So I'm not just a Pollyanna. We're already seeing some early signs of advances in treating human brain disorders. Right now with deep brain stimulators, much more sophisticated ones, that these things have been around for decades, but now actually by recording activity, by identifying biomarkers and being able to use that in real time to make better treatments for patients. We're seeing that already. I can only imagine once we have access to cell types based on the molecular information that Honghui was describing, with the tools that we can use to modulate activity, including with gene editing. I think the sky is really the limit, but we just have to keep working at it to get there. I think we'll be surprised in the next 10 years. We'll look back to see how far we've come.
Honghui, what are your thoughts?
I don't have much to add. I think as John said really well. Yeah, I agree.
I know that one of the things that's been most exciting looking at the field is not only the complexity of data, but the cost that it takes to get that data. But I guess one of the things that's stymied or is perhaps that turning point is transferring data into knowledge. And so I know that that's why open science is such an importance here, because the last thing you want to do is try to reproduce data sets. Is there a discussion, or where do you see being able to bring these streams of data together in a more comprehensive way, or even adding other flavors of data? I know there's deep phenotyping. I know that there's talks about how to maybe be more comprehensive, integrative, with all these different data points to look at it in a more systems way or a more holistic way. What are your thoughts there, and what's the potential, as well as maybe some of the challenges that we have?
Yeah, the short answer is yes. It's extremely important. Brain is really doing all we can to not just promote, but to enable rational and comprehensive access to the resources that we are helping to support out there in the field. It's going to take a lot more effort. I mean, we're trying to get ahead of the curve. Part of the challenge is that groups like Honghui's group and others at Allen are generating data faster than could have been believed just a few years ago. So we're literally building the plane as we fly it. But I think we're on the right approach. Brain has helped establish data coordination networks. It's going to be important not just for these molecular analyses, but for analyses across the neuroscience domain. we're trying to set this up as a framework that can be used across the board, especially as researchers are able to start using these tools to examine directly disease models, human disease models. So it's a big lift. We do have the benefit of incredible advances in data science and in computer science, and really the interest of data scientists and computer scientists and engineers in these problems. I mean, you know, great engineers love great problems to solve, and that's how we get it done. I do recognize this is a big challenge, but I think it's one that, you know, we if we set our minds to it, we can certainly make a lot of headway on.
Yeah, I just wanted to add that the Allen Institute has this open science mission from day one to make our data publicly available as a resource to the community. And we'll continue with that mission, absolutely. But we also think that this open science has to be widely adopted by the field. As John was saying, I think one of the main barriers to that is to overcome is the issue of credit assigning, right? How do you, who gets it first? And I think some kind of a mentality needs to be changed in the scientific community into how to reward people, how to promote people. I think, sharing data, sharing ideas should be something much more important now than just personal accomplishment, being the first in doing something. But of course, that's difficult. There needs to be mechanisms created that support, promote, encourage open science.
I mean, it's been exciting to see the discussion today, both from our speakers and even in our dialogue, range from talking about the most basic of science to really translational impacts. And in some cases, the dots are quite connected. It's amazing with that foundation, what can happen. So I'm curious as to when you look at the pipeline, or when you look at the broad spectrum of the most basic of information and all the different steps along the way to maybe translation, do you see that actually as shortening, given that foundation is building to such an extent, and really you can leapfrog based on all of the work that's been done currently and the rate of which work is being done now? Do you see that yourselves, or what are your concerns or excitement about the process itself?
Yeah, I think we're beginning to see that already, an acceleration. I think some of the most exciting fields in biology, when I look across the field, they're observing some version of Moore's law. There's certainly an acceleration, there's some kind of a doubling over some period of time. And I think that reflects the role that technology does play in enabling these discoveries. I think in terms of therapies, if you take, for example, the field of gene therapy, we're seeing some big breakthroughs. I mean, I think advances require disruptions, but then after those disruptions, the big question is, how does one democratize these innovations? And a democratization for me has become a personal and I think institutional mantra. And that these discoveries and technology innovations that we make, we need to disseminate and democratize them. And one way to do that is to find ways of basically taking technologies and learning how to apply them across different modalities. And if we can build modular toolkits, if we can take know-how in one area and apply it to another, be it from basic science to some disease application or cross-disease modalities, I think this will be a big win, and it's certainly going to be doable. I mean, we can certainly focus on areas where development in one area we think can be applied to multiple different areas. And I think this is really the beauty of what we're doing here in neuroscience with partners like the Allen Institute. I think there's really a potential to accelerate a lot in the next 5-10 years.
Ankwi, what are your thoughts, given the arc that your work has taken?
I don't have any additional thoughts. I think John said it really well.
I know it has been exciting to see all of this move forward, and it does seem to be at a dizzying speed almost. So I almost want to put a stake in the ground and say 5 years from now, I want to repeat this symposium and have the same people back to see how quickly things may have changed or how the view may have changed. Because I think even in a short timeline, 5 years, the landscape can dramatically transform. And it's exciting to say that. At the same time, it's a known unknown out there that we're all facing. So I'd like to leave each of you some time to have some closing thoughts as to, I know the time has gone quickly, as to your views, the state of brain health, or any other thing that you want to touch on that we haven't yet in regards to this topic. And maybe since John has been taking the lead role, we'll go to Hong Kui first.
Please.
What are your thoughts? What are closing thoughts, takeaways you'd like to leave people with today?
I think some of the main themes are the ones that we have already talked about. I think technology development, as John was saying, is incredibly important, and it's always transformative. It always brings breakthrough. The second point is multidisciplinary collaboration on open science is critical for the field going forward. Let's see. The third point is that I think that the field really needs to think boldly, new ideas, exploring new directions. That will always bring fresh perspectives, new insights, and really opens the door. I think new thinking, bold thinking, together with technology development will open doors for new frontiers. And the final point, specific point I just want to make is the synergy within this particular event, this webinar series. I think brain does not work in isolation. It works in its environment. Neuronal, non-neuronal interaction is, and brain-body interaction is really, really important. And it's an area that has been ignored by a lot of neurocentric people. So I really love to hear all the talks that we had today. Just really, again, it's just a way of bringing different perspectives into looking at the whole system together. Yeah, so those are my thoughts.
Thank you. John, what are your closing thoughts?
Yeah, these are great thoughts. I really appreciate them. I think what we've seen here today and what we've seen behind driving many of the great discoveries, not just here, but also across the field or fields of science, is the importance of curiosity-driven research based on creativity and, of course, being enabled by technology. I think the technology enables these ideas to lead to great discoveries. And it always reminds me to mention that chance does favor the prepared mind. We see this time and again. We don't always know when the next big breakthrough is going to come, but you have to recognize it when it does come. And the really transforming scientists and their discoveries came about because it almost seems they could have anticipated it just as or before it happened, or they actually made those opportunities happen. So I think there's just so many different possibilities. Technology plays a huge role in allowing us to ask questions we couldn't dream of before, but we really have to prepare our minds to seize those opportunities, to really make the big discoveries and to translate them into products and things and ideas that can really help the human condition. I think it's a great time to be in science. I know that, with COVID and all, a lot of us are kind of feeling down. We will get through this in large part because of the creative and resilient spirit of humans. But really, I can't imagine a better time to be in science. There's so much to do. There's so many tools at our disposal. There's so many questions we have left to ask. It's just a great time to be doing this.
Well, I can't agree more. I would totally agree. I think both of you stressing the creativity, the bold ideas, the smart luck or the prepared mind. All of these are definitely things that we're definitely at an inflection point. I mean, one of the reasons that our founder, Paul Allen, was so excited about the area of biology is he saw it as the next frontier, truly the next frontier. And this is someone who already set a frontier. So it is exciting to see this go forward. It is exciting to see the potential that we talked about today and hear your thoughts as well. And we may contact you again in 5 years to revisit this and do another retrospective, because I think things, as you said, are going to be moving fast. So thank you so much for participating and giving us these looks into the future. And for all of our attendees as well, thank you so much for the Q&A. Thank you so much for participating in chat. Please feel free to look to our website for future events, and all the talks you've seen or you may have missed if you'd had to step out will be available on YouTube. So please feel free to look at them and stay engaged, stay ready for the future, and think boldly. With that, thanks again to our presenters, and thanks to everyone.
Thanks very much.
Thank you very much.
Goodbye.
Bye.
Bye, thanks.